Transposon mutagenesis. Transposons are mobile DNA segments that can randomly move and insert themselves in new positions of the genome. They are found in both prokaryotes and eukaryotes and can cause chromosomal rearrangements or affect gene expression if inserted into the coding region of genes. Transposons exhibit a much higher frequency of mutation and chemical mutagenesis and find various applications in molecular biology, including the introduction of selectable marker genes in recombinant strains, identifying oncogenes, etc. In bacteria, the DNA that is inserted into the genome is called an insertion sequence. A typical insertion sequence is presented schematically in figure 3.44. It mainly consists of a gene that encodes transposase, the enzyme responsible for the transposition, flanked by two inverted repeats, which are similar in sequence but oriented in opposite directions. Figure 3.44 general structure of a bacterial sequence. The major portion of the sequence encodes the transposase enzyme. Its element is also flanked by very short direct repeat sequences, 5 to 11 BP long, black boxes, which are not inserted during transposition. Eukaryotic transposons are also found to carry such sequences equivalent to bacterial elements and were first identified by Barbara McClintock in maize kernels as AC elements. The P element in Drosophila was also identified as DNA transposon. RNA transcription. Cells of our body possess similar genetic makeup and yet get differentiated into distinct cell types that perform entirely different functions. For example, adipocytes are fuel tanks that store lipids to combat energy insufficiency. Cardiomyocytes contract to enable the heart to pump blood to all parts of the body. Pancreatic cells secrete insulin, and the renal cells that filter blood to remove impurities and excess water. How does this happen? The answer lies in the specificity of a unique set of genes that get turned on, or often these cell types, which define their respective biological roles. This transfer of genetic information from the DNA sequence is set in motion through the synthesis of ribonucleic acid (RNA), a process called transcription. For several decades after the discovery of nucleic acids in the year 1868, RNA was considered simply a mediator between DNA and protein. It was, however, only recently that the RNA was recognized as an essential functional entity that is central to governing different mechanisms in a cell. RNA is involved almost everywhere in a cell, carrying out a wide range of functions beginning from translation of genetic information to catalysis of chemical reactions and regulation of activity of protein genes during cellular growth, division, differentiation, response to external stimuli, adaptation to changing environment, or even at death. It functions at all levels of the organization. It can interact with either DNA or another RNA through complementary base pairing and can also bind to a specific protein. Errors or changes in synthesis, processing, or stability of RNA have been well established to be the prime cause of several known human diseases. It is therefore important to understand the basic principles of transcription and its downstream mechanisms that convert the inactive primary RNA into a functional molecule. Experimental evidence on the existence of RNAs. Between the years 1956 and 1958, Elliot Kenbogen and his colleague Lazarus Astrachan, while working at the Oak Ridge National Laboratory in Tennessee, first observed and reported that RNA is synthesized as a preliminary step during the process of synthesis of bacteriophage proteins in E. coli upon phage infection. They used a radiolabeled isotope of phosphorus 32P to track the production of newly synthesized RNA and observed that its composition was very different from that of bacterial RNA but resembled closely to phage DNA. This was what later came to be known as messenger RNA, mRNA. This experiment implied that phage mRNA could get translated into host bacterium by using host ribosomes and indicated that ribosomes are non-specific workbenches for protein synthesis. This hypothesis was tested in 1961 by Sidney Brenner, Francois Jacob, and Matthew Meselson in an elevated experiment where they radiolabeled ribosomes of uninfected E. coli and allowed phage infection to happen in the presence of radioactive RNA precursors. They observed that bacterial ribosomes which were present before infection were the ones that exhibited synthesis of phage proteins. These findings reinforced that ribosomes are non-specific, and another type of RNA directs the synthesis of phage proteins in E. coli. Saul Spiegelman and his team extracted 32P labeled phage RNA from bacteria following phage infection in a parallel independent experiment. They noticed that the RNA hybridized only to phage DNA and not to bacterial DNA. These initial experiments established the presence of mRNA as an intermediary molecule to translate genetic information into proteins. It was finally in the latter half of the year 1961 when this concept was formally put forward by Francois Jacob and Jacques Monod as a part of their findings elucidating the genetic control of enzyme and virus synthesis. Afterwards, they were awarded the Nobel Prize in Physiology or Medicine in 1965. Classes of RNA. RNA was previously thought to exist as only three major types: mRNA, messenger RNA, rRNA, ribosomal RNA, and ternary transfer RNA, with a sole motive to synthesize proteins. Extensive research efforts in the past couple of years have revealed the presence of several other RNA types that are non-protein coding. For a long time, these non-coding RNAs were assumed to be uninvolved in gene expression and therefore referred to as junk. It was in September 2012 when an enormous hyperopic project called the Encyclopedia of DNA Elements ENCODE delineated the functional role of non-coding elements in the human genome and described how extremely important these elements are for the normal functioning of cells. Constituting about 60% of the total RNA, non-coding RNAs control gene expression by binding to proteins or other molecules to turn genes on or off, almost always without affecting the associated DNA sequence. All the common RNA types that are synthesized endogenously in cells and their prime functions are listed in table 3.2 types of RNA primary functions. Messenger RNA, mRNA. Encodes protein. Ribosomal RNA, rRNA. Translation of mRNA. Transfer RNA, TRNA. Transfers amino acids to the growing peptide chain. During translation. Small interfering RNA, siRNA. Gene expression regulation. Small nuclear RNA, snRNA. Splicing. Small nucleolar RNA, SNORNA nucleotide. Modification of RNAs. MicroRNA, mRNA, gene expression regulation. Circular RNA, circRNA. Gene expression regulation. Purine interacting RNA, purina transposon defense. Types of RNA primary functions. Ribonuclease P, RNase P. Catalysis A. Maturation of ternate ribozyme. Telomerase RNA component TRK maintains telomeres during DNA. Replication. Cisnatural. Antisense. Transcript cisnat gene expression regulation. Long non-coding RNA, LNC-RNA, gene expression regulation. Table 3.2 commonly synthesized RNAs in the cells. The human genome encodes nearly 20,000 protein coding genes, which represent less than 3% of the total genomic sequence, and the majority of the remaining genome is transcribed into non-coding RNAs. With the increase in physiological complexity during evolution from prokaryotes to eukaryotes, there has been a steady increase in the amount of non-coding genome, figure 3.45. This proportion of the non-coding genome is what makes us human. Figure 3.45 percentage of non-coding genome in comparison to the total genome of an organism. The content of the non-coding genome rises with an increase in the complexity of an organism. Besides function, various RNA classes differ remarkably within an organism in terms of their size, stability, and abundance. Table 3.3. RNA types. Total number in humans. Genes transcripts. Overview of transcription. Transcription is mediated by a DNA-dependent RNA polymerase, RNAP, an enzyme that attaches ribonucleotides in a sequence complementary to the template strand of DNA. Unlike DNA polymerases, RNA polymerase does not require a primer and lack proofreading activity. To begin transcription, a genomic region of 20 to 30 base pairs is melted to fit the RNAP complex, forming a transcription bubble. The template DNA strand is read in 3 feet to 5 feet, and the synthesis of RNA occurs in the 5 feet to 3 feet direction. Genes can be transcribed from either of the two strands of DNA, with the direction of RNA synthesis always being in the 5 feet to 3 feet direction. So, the sequence of synthesized RNA is the same as the non-template strand of DNA, i.e., called the coding strand with thymine being replaced by uracil in RNA. Figure 3.46. Figure 3.46 transcription bubble. The template strand is read in the 3 feet right pointing arrow 5 direction. RNA is produced in a 5 feet right pointing arrow 3 direction with a sequence like non-template DNA, which constitutes the coding strand of the transcribed gene. For a given gene, only one of the two DNA strands can serve as a template. The transcribed DNA region between initiation and termination is called the transcription unit. Figure 3.47. Transcription start site TSS is specified as +1 position on the DNA sequence, and the successive nucleotides are numbered accordingly as the transcription proceeds downstream. The nucleotide upstream of TSS is denoted as -1. Figure 3.47 transcription from 38 srRNA gene. Visualized by electron microscopy as seen in Miller spreads of nuclei from salamander oocytes. RNA polymerase. Prokaryotic RNA polymerase. Nearly all the RNA in bacterium is synthesized by a single species of RNA polymerase, RNAP. The crystal structure of the bacterial RNAP core enzyme was first deduced from the thermophilic bacterium T. aquaticus. Bacterial RNAP occurs in two forms, core and holoenzyme. The core complex is 400 kDa and consists of two subunits, identical B and B units, and in W subunit. Figure 3.48. The two A and W subunits aid in the assembly of the enzyme. The B subunit binds Mg2 plus ions and is responsible for polymerase activity, and B helps in binding to the template strand. This core complex of 5 subunits lacks specificity, i.e., it is unable to spot the promoter element in the DNA sequence. This inability of the enzyme is resolved by a sigma factor, sigma, that helps in promoter recognition, melting of template DNA, promoter escape, and clearance figure 3.49. The sigma associates with the core enzyme complex to form the holoenzyme figure 3.48. Multiple distinct sigma factors recognize different groups of genes, amongst which sigma 70 effectively transcribes nearly all housekeeping genes during exponential growth. Figure 3.48 bacterial RNA polymerase holoenzyme. Figure 3.49 promoter recognition by sigma factor. Sigma factor binds to the -35 and -10 regions facilitating the attachment of the RNAP core complex to initiate transcription. Figure 3.50 different types of RNA polymerase III promoters. The type 1 promoter of the xenophosylate of 5S RNA gene consists of an internal control region ICR, which can be subdivided into a box plus 50 to plus 60 intermediate element, i.e., plus 67 to plus 72, and C box plus 80 to plus 90. The type 2 promoter of the X-late external gene consists of an A box plus 8 to plus 19 and AB box plus 52 to plus 62. The type 3 promoter of the Homo sapiens U6S RNA gene consists of a distal sequence element DSE, -215 to -240, that enhances transcription, and a core promoter composed of a proximal sequence element PSE, -65 to -48, and a top box -32 to -25. The Saccharomyces cerevisiae promoter is a hybrid promoter consisting of a top box -30 to -23, and A box +21 to +31, and AB box located downstream of the U6 coding region from +234 to +244 relative to the start site of transcription. The holoenzyme preserves the Crab claw structure of the core enzyme. Association of sigma 70 subunit and a larger part of non-conserved internal domain of B subunit in the complex significantly enhances the grip of B subunit, thereby making the Crab claw even more compact. In the holoenzyme, 2 N-terminal domains of sigma form A, B, like structure adjacent to the mouth of the upstream DNA binding channel, constituting the active site pocket of RNAP. The C-terminal domain of sigma falls close to the opening of the RNA exit channel. The extended linker arm forms a hairpin that overhangs into the active site pocket and stretches across the RNA exit channel to link the N and C-terminal domains. Eukaryotic RNA polymerase. RNA polymerases in eukaryotes share a good deal of sequence and structural similarity with prokaryotic RNA polymerases. However, eukaryotic cells have 3 distinct types of RNA polymerases wherein each polymerase class transcribes a certain set of genes, as listed below in table 3.4. RNA polymerases transcribe genes like rRNA, 2 protein mRNA and all non-coding RNA 3 terna and 5 srRNA. Table 3.4 eukaryotic RNA polymerases and their transcribed genes. Figure 3.51 eukaryotic RNA polymerases I, 2, and 3. Pol I comprise 14 subunits. Pol II has 12 subunits, PDB1 WCM, and Pol III contains 17 subunits. Each type of eukaryotic RNAP is composed of 2 large subunits that form the active site of the enzyme, i.e., where the transcription happens. These large subunits are accompanied by multiple small subunits and together confer specificity to different RNAP types. Figure 3.51. The 2 large subunits of RNAP2 contain 11 cleft loops, 7 and 4, respectively, that are essential for polymerase functionality. The clefts vary in size from 8 to 35 amino acid residues in Saccharomyces cerevisiae, table 3.5, and are essentially oriented towards the active site in the actual protein complex. These regions are dispersed all over the primary sequence of the large subunits and lack physical interactions with the non-neighboring loci in free RNAPs. Nevertheless, these regions become systematically arranged when an RNAP begins active transcription. Subunits cleft. Loops. Location. Amino acids functions. Largest zipper 39 to 51 maintains upstream terminal of the transcription bubble. Lid 248 to 260 binds and stabilizes upstream segment of DNA RNA hybrid and partakes in the foundation of RNA exit cleft by interacting with the flap loop. Subunits cleft. Loops. Location. Amino acids functions. Rudder 304 to 324 binds and stabilizes upstream segment of DNA RNA hybrid. Switch 2328 346 pairs the presence of nucleic acid in the pre-initiation complex to shutting off the clamp. Via interacting with the bridge helix in the template strand of DNA. Bridge 811 to 845 an alpha helix that navigates the active core located immediately downstream of catalytic. Mg2 plus and interacts with switch regions in elongation complex. Trigger loop. 1084 1091 crucial for transcription. Switch 11384 1406 pairs the presence of nucleic acid in the pre-initiation complex to shutting off the clamp. Via interacting with the bridge helix in the template strand of DNA. Second largest fork. Loop 1466 478 binds and stabilizes upstream segment of DNA RNA hybrid. Fork. Loop 250512 enables split of template and non-template strand immediately downstream of the bridge helix and catalytic Mg2 plus. Flap loop 920 to 933 involved in foundation of RNA exit cleft by interacting with the lid. Cleft loops are highly conserved regions of RNAPs but differ significantly in terms of length of sequences and amino acid substitution rates, especially near the enzyme's catalytic site, thus leading to polymerase-specific transcription of eukaryotic genes. Transcription in prokaryotes. Prokaryotes do not feature membrane-bound nuclei, so the process of transcription, translation, and mRNA degradation all occur at the same time. Prokaryotic RNAP often transcribes more than one gene in a single go, leading to the synthesis of polycistronic mRNAs that code for more than one protein. The transcription involves 3 major phases: initiation, elongation, and termination, followed by the release of the core enzyme and nascent RNA chain. Figure 3.52. Figure 3.52 transcription cycle in bacteria. RNAP binds to promoter element on DNA sequence through the help of sigma factor and dissolves the base pairing between the 2 DNA strands to form a pre-initiation complex. After the phosphodiester bonding of the first RNA dinucleotide, the sigma factor dissociates. Successive residues are added to the 3' end of the grown RNA chain. After reaching the first 10 to 20 nucleotides of synthesized RNA, RNAP undergoes a conformational change to move away from the promoter element promoter clearance. Once the RNAP core enzyme reaches the termination sequence in the DNA strand, newly formed RNA and RNAP are released from the template. Initiation and elongation. The RNA polymerase binds to multiple regions on the DNA with low affinity and scans it at the rate that is greater than or equal to 1000 base pairs per second till it finds the specific promoter where the transcription must be initiated. This holoenzyme binds to the promoter of such gene, forming a pre-initiation complex PIC. This produces a conformational change in the RNAP, the prokaryotic promoter transcription bubble. As the DNA unfolds, supercoils are generated that are smoothed by DNA topoisomerases in both upstream and downstream of the transcription bubble. The nucleotide present at TSS, i.e., +1 forms the 5' nucleotide of the future mRNA transcript. Figure 3.50. RNA polymerase catalysis is the formation of phosphodiester bonds to incorporate ribonucleotides in an order complementary to the downstream DNA sequence dictated by the Watson-Crick base pairing rules. Is constituted of certain conserved sequence elements that are identified by sigma factors. A conserved sequence 5' -3 TDGAGA located 35 bases upstream of TSS is the first one to be recognized and contacted by the holoenzyme. Figure 3.53. After binding, the polymerase moves along the sequence in the downstream direction and encounters another conserved element called top box 5' -3 TAT housed at 10 bases upstream of TSS. Figure 3.53 and results in the unwinding of DNA approximately 14 bases forming. Figure 3.53 the promoter element in prokaryotes. The sigma subunit of prokaryotic RNAP identifies the conserved sequences located upstream of TSS in the promoter region. Subsequent to the integration of initial 10 to 20 nucleotides in the nascent RNA chain, the polymerase stalls, called polymerase pausing or promoter clearance, and undergoes a second conformational change to move away from the promoter to go in the elongation stage. This transition is a crucial step to regulate gene expression. Termination. RNA synthesis in prokaryotes is completed when RNAP either arrives at a termination signal in DNA sequence or is facilitated by a protein called Rho. Rho independent termination. A termination signal sequence in DNA transcribes a self-complementary GC-rich sequence in nascent RNA followed by a stretch of uracils at 3' end. This induces the synthesized RNA to fold back on itself, creating a stable hairpin structure. The subsequent uracil towards the 3' end of the mRNA form relatively weaker bonds with complementary adenine residues in the template DNA. This event along with the stalled RNAP generates sufficient instability for the core enzyme to lift off and let go the new mRNA molecule. Figure 3.54. Figure 3.54 Rho independent termination of transcription in bacteria. Upon reaching the termination sequence in template DNA, the RNA chain folds back on itself to create a stable hairpin structure. This weakens the binding of RNAP, resulting in its release in the RNA chain. Rho dependent termination. Rho factors in activates with helicase activity and locates the signal sequence C-rich site, Rho recognition site, near the 3' end of RNA. Rho moves along the RNA towards the termination site where the paused RNA polymerase is seated. Figure 3.55. Its helicase activity disrupts the RNA-DNA hybrid helix releasing the RNA chain. Also, the polymerase detaches from the DNA template. Since transcription and translation happen simultaneously in bacteria, the Rho protein does not bind to the RNAs being translated. Instead, it binds to such RNAs after translation when the transcription is still on. Figure 3.55 Rho dependent termination of prokaryotic transcription. Rho attaches to a recognition sequence in synthesized RNA and unwinds the RNA-DNA hybrid by its helicase activity to set free the synthesized RNA. NUSA and NUSTR are major regulators of bacterial transcription elongation, which act either in concert or antagonistically. Both bind to RNA polymerase RNAP, regulating causing as well as intrinsic and Rho dependent termination. Figure 3.56. Figure 3.56 Possible functions of NUSC interaction in transcription regulation. First, the NUSA interaction may play a role in the recruitment of NUSC to the tech. Second, it may provide a long linker between RNAP and the ribosome consisting of NUSA and NUSC, which would allow resynchronization of transcription, translation coupling. Third, the NUSA interaction may release the autoinhibition of NUSA, allowing constitutive binding of NUSA SKK to RNA, so that recruitment sites were lower blocked and Rho dependent termination is decreased. Fourth, NUSA AR II may obstruct NUSC NTD and intrinsic termination sites, facilitating the release of nucleic acids and enhancing intrinsic termination. Transcription in eukaryotes. As anticipated, eukaryotic transcription is far more complex than prokaryotes. In addition to the presence of specific RNA polymerases for a different set of genes in eukaryotes, the chromatin must be remodeled before transcription. The nuclear DNA is packaged over histone octamers called nucleosomes, and it must be made accessible to the transcriptional machinery. Figure 3.57. Actively transcribed regions of the genome are loosely packed euchromatin and therefore conveniently available for transcription. On the contrary, the inactive genome is densely packed heterochromatin. A common mechanism that remodels chromatin is the modification of residual chains of histone proteins, acetylation, ubiquitination, methylation, etc. Chromatin remodeling is a major aspect that regulates gene expression under diverse circumstances. Figure 3.57 Chromatin packaging and accessibility to transcriptional machinery. Eukaryotic transcription is principally governed by the chromatin state of the gene where the transcription needs to be initiated. Loosely packed chromatin or euchromatin enables the binding of the RNAP complex in contrast to heterochromatin. In humans, the TATA box consensus sequence 5'3", TATA AA, is located 25 to 30 bp upstream of a gene's TSS and is bound by a 34 kDa TATA binding protein TBP. Figure 3.58. A second conserved sequence is known as a COT box. Is present 70 to 80 bases upstream of TSS. Certain constitutively expressed genes lack a CAAT box but then contain a GC-rich region GC box. The genes that lack the TATA box harbor additional regulatory regions initiator sequence INR and or downstream promoter element DPD. The INR spans the TSS position -3 to +5 and DPD is located 25 bases downstream of TSS. All these regions represent the cis-acting or proximal elements of a gene in mammals and serve as seats for transcriptional machinery and the associated proteins. Besides cis elements, the eukaryotic genome also features several distal elements, enhancers and repressors that regulate a gene's expression, i.e., increase or decrease its transcription by binding various TFs. Enhancers and repressors can be present in any orientation upstream of TSS, downstream of TSS, or even within the transcribed region of the gene itself. A mutation or a nucleotide polymorphism in any of these crucial regulatory elements, promoter proximal or distal, can disrupt the gene's function and end up in a disease phenotype. Figure 3.58 Central elements of the promoter of a eukaryotic gene. It constitutes a set of proximal sequences CAAT box and or GC box and distal sequences enhancers and repressors that govern a gene's expression. Genes lacking the TATA box possess initiator sequence INR and or downstream promoter element DPD within the transcribed region of the gene. Like prokaryotes, RNA polymerase II in eukaryotes is unable to identify or bind to the promoter by itself. It requires the involvement of several additional proteins called transcription factors TS, which are encoded by different genes to form the pre-initiation complex PIC. The key TFs, namely IID, IIB, IIF, E, and IIH, help in positioning RNAP2 at a gene's promoter, so they are also known as general initiation factors or GIFS table 3.6. TAF-TBP associated factor, GTF general transcription factor, TBP TATA box binding protein table 3.6 general transcription initiation factors from humans. The complex formed by TBP and associated proteins is referred to as TFIID, and its binding to the TATA box signifies the initiation of transcription. The binding of human TFIID to genomic DNA was originally described with the major late promoter of adenovirus ADMLP. DNA site footprinting of ADMLP and other human gene promoters highlighted sequence-specific contacts between human TBP and the TATA box, principally through the TBP subunit of TFIID. Figure 3.59. The 3D structure of TBP resembles a horse's saddle, which helps TBP to provide a stable platform to bind other TFs at its convex upper surface and simultaneously support DNA binding at its concave underside. TBP consists of two pseudo-identical domains conforming to the two direct repeats present in the preserved region of TBP. The binding of TBP bends the DNA by 100 degrees, which further facilitates the assembly of the rest of the transcription machinery. PIC assembly about this bend leads to a more condensed multi-protein DNA complex, and it also helps to fetch remotely bound transcriptional activators near to the core promoter element to interact with PIC. Figure 3.59 Binding of TBP bends the DNA to facilitate a compact interaction of TFs and bring the remotely bound TFs close to PIC. Binding of either TFIID or TBP at the TATA box prevents the packaging of the gene's core promoter with histones proteins that together constitute a nucleosome. Alternatively, the core promoter elements packaged by nucleosomes do not allow TFIID or TBP to bind at the TATA box, resulting in a repressed expression of the respective gene. Such chromatin-mediated repression of transcription is mended within a cell by certain ATP-dependent macromolecular machines, e.g., the SWISNF complex that modify the chromatin near the core promoter. TFIID is a subsequent gift to join the PIC. The basic cleft of TFIID clamps to the acidic C-terminal loop of TBP and interacts with its phosphoribose backbone at the TATA box to form the ternary complex TFIID-TBP DNA. The resultant TFIID-TBP DNA codium is then identified by a complex of RNAP2 and TFIIF, followed by TFIE and TFIIH. TFIIF is a heterodimer of two distinct subunits, masses of 30 kDa and 74 kDa, respectively, and has a unique ability to form an extremely stable complex with RNAP2 known as polyf. Within the PIC, TFIIF localizes to the core promoter between the 3' end of the TATA element and TSS. TFIE is in a 2v2 heterotetramer of subunits with respective masses of 34 kDa and 56 kDa, and it positions at the same site as TFIF. TFIIH, however, is a much larger multi-protein assembly composed of 9 different subunits with masses between 39 to 89 kDa. TFIIH distinctively stages various catalytic processes like DNA-dependent ATPase, ATP-dependent DNA helicase, and a serine threonine kinase phosphorylate the C-terminal domain of the large subunit of RNAP2. TFIIH is also known to function in DNA repair. Its subunits ERCC2 and ERCC3 are important components of the repair machinery of DNA excision. TFIIA was previously thought to be crucial for gene transcription. However, subsequent to an extensive molecular characterization of the genes that encode subunits of TFIIA, it is now well established that TFIIA is a coactivator that enables the regulation of RNAP2 transcription. During PIC assembly, TFIIA combined with and stabilize the TFIID DNA or TFIIB to the DNA complexes to augment the stimulatory effects of transcriptional activators and protect the complex from the harmful effects of inhibitory cofactors. The TPA uses a heterodimer consisting of 14 kDa and 32 kDa masses. It identifies the internal loop of TBP and interacts with the phospholipase backbone upstream of the TATA box on the DNA helical site, which is opposite to where the TFIIB binds. This entire transcription machinery occupies almost 60 nucleotides extending from position -30 to +30 with respect to the TSS. Figure 3.60 Basic transcriptional machinery in eukaryotes. TFIID binds to the TATA box and is followed by the binding of TFIIB, TFIIF RNAP2, TFIE, and TFIIH. TFIIA is not an initiation factor, but a cofactor that binds early during PIC assembly to regulate RNAP2 transcription. Once the PIC is fully assembled and nucleoside triphosphates are present, the strands of DNA separate at the TSS, forming an open complex. The C-terminal domain of the large subunit of RNAP2 is phosphorylated, and the transcription enters the elongation phase. To promote reinitiation of transcription by RNAP2, TFIID continues to stick to the core promoter. As the core promoter binding by TBP is a slow and energy-demanding process, TFIID remains stably bound through multiple initiation events to facilitate successive rounds of transcription. The basal transcription by TBP is augmented by the actions of specific proteins called activators. For instance, SP1, MyoD, and NF1 protein families bind to the GC box. CBBP and NFY bind to the CAAT box to increase transcription of its downstream genes. The signals of these activators bound to distant regulatory regions are transmitted to the transcriptional machinery by a large multi-protein complex called mediator. In yeast, Schizosaccharomyces samba, mediator is formed of 19 subunits and is nearly 0.8 MDA in size in contrast to 21 subunits in Saccharomyces cerevisiae. In humans, the mediator complex is 1.4 MDA in size and composed of 26 subunits, of which 5 subunits med23, med25, med26, med28, and med30 are mesozoan specific. A mediator complex contains 4 modules: head, middle, tail, and CDK8 kinase. It is recruited to the enhancer regions by its interactions with TFs, especially through the tail module. The mediator then transiently contacts the transcriptional machinery assembled at the core promoter element. This transition is accompanied by the 3' end of mRNA. The primary transcript is cleaved by RNA endonucleases at about 15 nucleotides of the eukaryotic cleavage and polyadenylation signal, AAUAA, and then processed further in the nucleus to generate a functional molecule. Dispersion of the CDK8 kinase module from the mediator. Thus, these mediator proteins act as transcriptional corregulators by activating or repressing gene expression. After transcribing the designated region and reaching the DNA sequence that encodes. Figure 3.61 The torpedo model for transcription termination by RNA polymerase II in the human B globin gene. B globin-free mRNA is subjected to a series of covalent modifications during its synthesis, capping, splicing, cleavage at the poly A site, polyadenylation, cleavage at the CoP C site, 5' right pointing arrow 3, degradation of downstream RNA and degradation of the RNA byproduct between the poly A and CoP C sites. CoP C is a ribozyme, 5' right pointing arrow 3, degradation of downstream RNA is carried out by XRN2 exonuclease, and both processes are required for termination. Drugs targeting RNA transcription. Lately, bacterial transcription has been a primary target for the development of antibacterial drugs. As of now, there are only 2 commercial antibiotics that target bacterial RNA polymerase: rifamycin and fidaxomicin bleepharmycin. Both these compounds inhibit the initiation of transcription in bacteria by binding to RNAP and preventing the formation of the first phosphodiester bond. Besides, an inhibitor of Rho factor, bicyclomycin, is also being used to promote growth in the animal feedstock. In eukaryotes, RNA synthesis is known to be inhibited or perturbed by the activity of several compounds: actinomycin D, A-amidin, DRB, flavopiridol, and tryptolite. Table 3.7. The action of actinomycin D on transcription inhibition is fast, but its selectivity for RNA polymerase II or 3 is poor. On the contrary, A-amidin is highly selective but acts slowly. The activity of DRB and flavopiridol is rapid and reversible, but several genes escape the inhibition of transcription. Tryptolite can trigger the degradation of RNA polymerase II and works speedily and selectively. ACTINOM. YCIN. D. A. Post-transcriptional processing of RNA is synthesized as pre-mRNA, which must be processed into a functionally active form. In prokaryotes, due to lack of cellular compartmentalization, primary mRNA transcripts require only allele processing and therefore begin to serve as templates for translation even when their transcription is ongoing. Prokaryotic tRNA, however, undergo rigorous processing. Nearly all eukaryotic RNAs, be it mRNA, mRNAs, lncRNAs, tRNAs, or rRNA, undergo extensive processing in the nucleus after being transcribed to serve their intended function. Also, the transport of the processed RNA from the nucleus to the cytoplasm is a highly synchronized and regulated mechanism. Transport RNA. Both prokaryotic and eukaryotic tRNAs are transcribed as lumbar units in the actual functional molecule. Figure 3.62. In eukaryotes, mature cytosolic tRNAs are synthesized by RNA polymerase III, RNAP3, in the nucleoplasm and range from 75 to 80 nucleotides in length. Any intron-specific sequences at both ends are cleaved by the nucleases. Certain nucleotides at the 5' end of nascent turn are cleaved by ribonucleoprotein endonuclease called ribonuclease P, RNase P. In E. coli, RNase P, i.e., a 14 kDa polypeptide and another 377 nucleotides long RNA called the M1 RNA are the first molecules which reveal that RNAs can perform catalytic functions. M1 RNA identifies and cleaves E. coli pre-tRNAs at higher cellular concentrations of Mg2+. RNase P incredibly catalyzes the cleavage by M1 RNA by allowing M1 to perform even at normal physiological levels of Mg2+. Not all the pre-tRNAs possess introns that will be spliced, but they all undergo 3 basic types of change during tRNA processing. Figure 3.62. Uracil residues at the 3' end of tRNA are replaced with a 3-nucleotide sequence CCA, which is found in all mature tRNAs. In addition, multiple bases undergo unusual modifications. The 2' group in the ribose sugar backbone of certain residues and the heterocyclic ring of purine bases are methylated. Specific uridines are modified to dihydrouridine, ribithiamidine, or pseudouridine residues. Figure 3.62 Processing of tyrosine tRNA following its synthesis. An intron colored in blue in the anticodon loop of immature tRNA is excised by splicing. Uracil residues at the 3' end are replaced by the CCA sequence shown in red. A nucleotide sequence shown in green at the 5' ends are cleaved by RNase P. Several bases are characteristically modified yellow. D equals dihydrouridine, Y equals pseudouridine. Ribosomal RNA. The 3 prokaryotic rRNAs 23S, 16S, and 5S, and eukaryotic rRNAs 28S, 18S, and 5.8S are synthesized as single pre-rRNA molecules. These single pre-rRNAs are cleaved by ribonucleases to generate right-sized units of RNA and transferred by endonucleases to generate designated rRNA species. Figure 3.63. Eukaryotic 5S rRNA is synthesized individually. Figure 3.63 Prokaryotic pre-rRNA. The 3 rRNAs 23S, 16S, and 5S are synthesized in continuation as single molecule and are finally cleaved by ribonucleases. In eukaryotes, nascent pre-rRNA transcripts are synthesized in the nucleolus and are instantly joined by specific proteins to form pre-ribonucleoprotein particles or pre-rRNPs. Selected proteins in pre-rRNPs remain bound with mature ribosomal subunits, whereas others confined to the nucleolus and aid in the assembly of the subunits. The largest pre-rRNP in the mammalian nuclei comprises of an intact 45S pre-rRNA molecule, which is cleaved to eventually generate the mature RNAs found in eukaryotic ribosomes. Figure 3.64. During processing, pre-rRNAs are also commonly modified by methylation of the 2' group of ribose sugars and modification of certain uridine residues to pseudouridine. The sites of 2' methylation and pseudouridine formation are decided by nearly 150 distinct small nucleolus restricted RNAs known as small nucleolar RNAs, SNORNAs. The SNORNAs hybridize transiently to parentage and associate with proteins to form SNORPs. In a SNORP called RNase MRP, the associated SNORNA is similar to the RNA of RNase P tRNA processing. RNase MRP catalysis the removal of transcribed spacer sequences from pre-rRNA. Figure 3.64b. Unlike other pre-rRNA genes, 5S rRNA is transcribed by RNAP3 outside of the nucleolus. Without any processing, nascent 5S rRNA straightaway moves to the nucleolus and combines with the 28S and 5.8S rRNAs and proteins to form large ribosomal subunits that are eventually transported through the nuclear pore complexes to the cytoplasm. Figure 3.64 Assembly of pre-rRNA in eukaryotes. Nucleolar proteins combine with 45S tRNA after their synthesis to form an 80S tRNB. 5S rRNA is synthesized in the nucleus but outside the nucleolus. B processing of 35S pre-rRNA nascent transcript in yeast S. cerevisiae. The transcribed spacer regions, beige colored, are removed during the processing to part the segments that form the mature 18S, 5.8S, and 25S rRNAs. Processing of mRNA. The mRNAs in most of the lncRNAs undergo rigorous processing before performing their respective intended roles to translate or regulate. Nearly all eukaryotic mRNAs and lncRNAs synthesized by RNA polymerase II are primarily heterogeneous RNAs, H and RNAs, which means that the coding sequences exons of transcripts are interrupted by non-coding sequences introns. These introns are removed, and the neighboring exons are joined together in a process called splicing. Splicing. A multi-unit complex called spliceosome transforms the primary H and RNA to the coding mRNA. It consists of 5 different uracil-rich small nuclear RNAs, U1, U2, U4, U5, and U6, a primary transcript, and a total of 60 to 70 RNPs. S and RNPs help in the correct alignment of neighboring introns and exons for splicing to proceed. A consensus sequence loopback called the branch site is located near the 3' end of the intron that should be excised. Figure 3.65. U1 attaches to the exon boundary at the 5' end of the intron. U2 binds at the branch site. The interactions of U4, U5, U6 within the S and RNP complex help in an approximation of 5' and 3' splice sites. The 2' group of an adenosine A residue in the branch site attacks the PO4. 3 group at the 5' end of this. Intron i.e. spliced donor site producing a cut to form a phosphodiester bond 5' minus 2' resulting in a lariat structure. Figure 3.65b. The cleavage is mediated by U2, U6 within the S and RNP complex. Following this, a second cut is created at the 3' intron exon junction, where the 3' group of the upstream exon attacks the 5' PO4. 3a group of the downstream exon intron junction liberating the intron lariat structure, which will get degraded later. The 2 exons are thus finally joined together. Figure 3.65 The process of splicing. The A residue in the branch site attacks the 5' end of the intron, which must be excised and forms a phosphodiester bond 5' minus 2' resulting in a lariat structure. The process is facilitated by a complex form of small nuclear unassociated proteins called spliceosome. Alternative splicing. Post-transcriptional processing, especially splicing, is a common mechanism to regulate gene expression. Faulty splicing can lead to the synthesis of wrong proteins or even truncation leading to cellular dysfunction and disease. Instead of always being spliced similarly, a process called alternative splicing enables a single mRNA species to splice differently under specific conditions for certain tissues and therefore synthesize different protein variants or isoforms possessing different functions or properties in the cell. This is executed by reorganizing the pattern of introns and exons that will be spliced to alter the coding sequence of mRNA. Figure 3.66 Alternative splicing. It leads to the synthesis of entirely different proteins, isoforms from the same gene. The synthesized protein isoforms vary in sequence, structure, and function. This cellular process is implemented in different tissues or under certain physiological conditions. Alternative splicing can be accomplished by any of the processes described in Figure 3.66. Up against the constitutively spliced primary mRNA transcript, sometimes one exon is included while the other is certainly excised mutually exclusive exons. An intron is retained when it is not spliced but is retained in the mature mRNA. Besides, there can be a change in conventional splice sites, alternative splice sites, leading to a variability in the length of 5' and 3' exons. Or in certain cases, there is a change in the TSS of a gene due to the use of alternate promoters resulting in the synthesis of a changed transcript. Figure 3.67 Different types of alternative splicing. Introns are depicted as a black line, and exons appear as red, blue, green, and pink boxes. The splicing event has been shown as a dotted line. Figure 3.68 Comparison of cis and trans splicing. In cis splicing, the U1S NRNP base pairs with the 5' splice site, and U2S NRNP base pairs with the branch point near the 3' splice site. The intron is excised, and the two exons are spliced together. In trans splicing, there is no 5' splice site on the pre-mRNA for U1S NRNP binding. Instead, the 5' splice site is provided by the donor SLS NRNP, which may base pair with a sequence in the outron, the region of the pre-mRNA between the 5' cap and the trans splice site. The SLX is then spliced to the first exon on the pre-mRNA. CBC, nuclear cap binding complex. Capping. The mRNA cap is a highly methylated modification of the 5' ends of RNA pol II transcribed RNA. It protects RNA from degradation, recruits complexes involved in RNA processing, export and translation initiation, and marks cellular mRNA as self to avoid recognition by the innate immune system. A 7-methyl-1-OC molecule is attached to the 5' end of mammalian mRNAs through a 5' minus 5' triphosphate linkage. This is the first step in RNA processing. The G-phosphate 5' triphosphate of pre-RNA is removed and replaced with guanosine monophosphate (GMP) by the enzyme guanylyl transferase in the nucleus. This guanine residue is methylated by the enzyme guanine 7-methyltransferase in the cytosol. The 5' capping is needed for cellular stability and efficient translation of RNA. The cap also prevents the 5' end of the RNA from being attacked by exonucleases. Figure 3.69 Capping of mRNA in eukaryotes. A 7-methyl-1-OC molecule is attached to the 5' end of mRNAs through a 5' minus 5' triphosphate linkage through the action of various enzymes. Polyadenylation. Long stretches of adenosine residues, poly A tails, are joined to the 3' end of all mRNAs and most valencinous in a process called polyadenylation. Poly A tails contribute to the stability of an RNA by protecting its 3' terminus from attack by exonucleases and is also required for its transport from the nucleus to the cytoplasm. Nearly 20 bases downstream of the recognition sequence AAUAA, an mRNA is cleaved, and the enzyme poly A polymerase incorporates a poly A tail that extends to as much as 200 adenosine residues. Figure 3.70. Figure 3.70 Polyadenylation. Upon reaching the poly signal sequence AAUAA, the nascent mRNA is cleaved, and a long stretch of adenosine residues is incorporated towards the 3' end by the enzyme poly A polymerase. Figure 3.71 Model for CSTF assembly, nuclear translocation, and polyadenylation activity. CSTF assembles in the cytoplasm mediated by the specific interaction of CSTF64 with CSTF77 via the hinge domain, left CSTF50 presumably associates at the same time. The assembled CSTF complex is then transported to the nucleus center mediated by the nuclear localization signal in CSTF77, NLS CSTF77. Finally, CSTF associates with CPSF through CSTF77 CPSF160 interaction, mediating recognition of authentic polyadenylation sites followed by cleavage and polyadenylation of the pre-mRNA. Processing of tRNA. Bacterial tRNAs are transcribed as pre-RNAs and become functional after a series of processing which includes kind events and chemical modifications. Most of the genes for tRNAs are randomly distributed around bacterial genomes. Some are distributed as tandem arrays in multi-unit transcription units, and some as infiltrators in the rRNA transcription units. For example, E. coli has 1 or 2 tRNA genes between the 16S and 23S genes in rRNA transcription units. The processing of all pre-tRNAs is similar but not identical. The precursor molecule with the tRNA sequence adopts a codally structure with additional 2 hairpin structures on either side of the tRNA. Figure 3.72. Ribonuclease E or F form a new 3' end by cutting just upstream of one of the hairpins. 7 nucleotides from this new 3' end are then turned by ribonuclease. D in the 5' end of mature tRNA is formed by ribonuclease P by cutting at the start of the cloverleaf. Ribonuclease D removes 2 more nucleotides and creates 3' end of mature tRNA, which ends with 5' CCA3. Some of the pre-tRNAs have this sequence, but some which are created by ribonuclease C lack the sequence and thus need to be synthesized by tRNA nucleotide transferase, which is a template time-dependent RNA polymerase. Most of the modifications are done on an already existing nucleotide in the transcript, but some modifications are done by cutting the existing nucleotide and replacing it with a new version, e.g., Yosine and Qosine. Approximately 1 in 10 nucleotides in tRNAs are found to be altered. The modifications help enzymes to identify individual tRNA to attach to particular amino acids and also enable one tRNA to recognize more than one codon. In eukaryotes, pre-tRNA modification involves precursor splicing with the help of unique nuclease and ligase activities. The excision of introns from tRNA precursors occurs in 2 stages. In stage 1, 2 cuts are made at the ends of the intron by a nuclear membrane-bound splicing endonuclease. Then, in stage 2, a splicing ligase joins the 2 halves of the tRNA to produce the mature form of the tRNA molecule. Nucleotide sequences have no role in the specificity of these reactions. The specificity lies in conserved 3-dimensional features of the tRNA precursors. Various enzymes like an RNase P, RNase C, RNase D, RNase E, RNase F play an important role in the formation of the codally structure of functional tRNA. Nucleotide modification is yet another process through which pre-tRNAs are bestowed with different chemical properties. Over 50 different chemical modifications are known in total. Some of the modifications are: 1. Methylation addition of 1 or more CH3 groups to the base or sugar. 2. Deamination removal of an amino NH2 group from the base. 3. Inosine a deaminated form of adenosine. 4. Sulfur replacement of oxygen with sulfur. 5. Base isomerization. Positions of atoms in the ring component of the base are changed, e.g., isomerization of uridine gives pseudouridine. 6. Double bond saturation. Converts double bond to a single bond, e.g., the conversion of uridine to dihydrouridine. 7. Nucleotide replacement substitution of an existing nucleotide with a new one, e.g., Qosine. Figure 3.72 Processing of pre-tRNAs. Processing of rRNA. Similar to bacterial tRNAs, the bacterial rRNAs are also first transcribed as pre-RNAs which become functional after a series of cutting events and chemical modifications. 3 different types of rRNAs based on sedimentation analysis are synthesized, i.e., 5S rRNA, 16S rRNA, and 23S rRNA. A single transcription unit has genes for all the 3 rRNAs, which also means that pre-RNA has copies of 5S rRNA, 16S rRNA, and also 23S rRNA. Figure 3.73. The cuts needed to release mature rRNAs are made by ribonucleases 3, P, and F. The specificity of cutting is decided by the base pairing between the ribonuclease and different parts of the pre-RNA. Ribonucleases M16, M23, and M5 then turn the cut end and finally form the mature, functional rRNAs. Figure 3.73 Pre-RNA processing. Source: Genome Third Edition by Tom Brown. Genes for all the 3 types of RNA and some genes for tRNA are present in the same tRNA. The cut to release mature 16S rRNA and 23S rRNA is made by ribonucleases 3, whereas ribonuclease P and F releases mature 5S rRNA. Trimming of the cut ends of 16S rRNA, 23S rRNA, and 5S rRNA is done by M16, M23, and M5, respectively. Chemical modifications are also observed in pre-RNAs as a part of processing in bacterial prokaryotic systems. rRNAs are modified in 2 ways. Methyl group is added to the 2-PO group on nucleotide sugars, and secondly, base isomerization, i.e., conversion of uridine to pseudouridine. All copies of an RNA are modified at the same position in different species. Similarities are seen even in bacteria and eukaryotes, with only differences in level of the complexity. rRNA with modified nucleotides play a role in the synthesis of peptide bonds. Modification involves direct recognition of the sequence to be modified by enzymes. In eukaryotes, the introns present in rRNA precursors are removed autocatalytically. It is unique in the sense that the reaction is intrinsic to the RNA molecules, and no protein enzyme is involved in it. This was first discovered in 1982 by Thomas Cech and his coworkers in Tetrahymen and Drosophila. This autocatalytic activity is shown by rRNA precursors of lower eukaryotes and rRNA, tRNA, and mRNA precursors in mitochondria and chloroplasts of many species. Their mechanism of autocatalysis is the same as that of Tetrahymen and Drosophila. Still, others follow the splicing mechanism of nuclear mRNA precursors but without the involvement of the spliceosome. The former mechanism of autocatalysis requires no external energy source and no protein catalytic activity. It only involves the transfer of a series of phosphodiester bonds without any bond to be lost or gained. Monovalent cation and a divalent cation along with guanine nucleoside or nucleotide with a free 3' group like GTP, GDP, GMP, or guanosine are the requirements of the reaction. Transfer of two phosphodiester bonds excises the intron, which gets recircularized by another phosphodiester bond transfer. The self-splicing resides within the intron structure itself, more properly in the secondary structure of the intron or of the RNA precursor. The secondary structure brings the reactive groups into close juxtaposition to allow the transfer of the phosphodiester bond. Splicing reactions are intermolecular in nature and do not depend on concentration. However, the excised introns should either be degraded rapidly or spliced rRNAs should be exported immediately to the cytoplasm so that the whole process remains irreversible. CTD of RNA polymerase II coordinates transcription and processing. Eukaryotes have three nuclear DNA-dependent RNA polymerases: RNAPs, RNAPI, RNAP2, and RNAP3. RNAP2, responsible for the synthesis of all mRNA as well as many non-coding RNAs, ncRNAs, consists of 12 polypeptides, of which RPB1, which possesses the enzyme's catalytic activity, is the largest. RPB1 also contains a C-terminal domain, CTD, composed of tandem heptid repeats that constitutes a unique feature of RNAP2 and distinguishes it from all other polymerases. The C-terminal domain, CTD, of the RNA polymerase II largest subunit consists of multiple heptid repeats, consensus tier 1 S2 Pro 3 THR 4 SR5 Pro 6 SR7, varying in number from 26 genes to 52 invertebrates. The CTD orchestrates multiple events during the transcription process. Genes transcribed by RNAP2 are bound dynamically by nucleosomes. The CTD, modulated by phosphorylation, provides a means to recruit histone modifiers and chromatin remodeling complexes to influence transcription initiation, elongation, and termination. Figure 3.74, Figure 3.76. Figure 3.74, dynamic modification of the CTD during the transcription cycle. At transcription initiation, CDK7 phosphorylates SR5 and SR7 residues. Later, during elongation, CDK9 phosphorylates SR2 and perhaps THR4, while the phosphate groups on SR5 and SR7 are gradually removed by phosphatases. For example, RTR1, likely indirectly with another phosphatase, and SSU72, with the aid of the prolysomerase PIN1, dephosphorylate SR5P early and late during elongation, respectively. SSU72 also dephosphorylates SR7P. CDK12 likely also contributes to SR2 phosphorylation during elongation of at least some genes. As RNAP2 nears termination, FCP1 dephosphorylates SR2P, regenerating unphosphorylated RNAP2 that can be recycled for another round of transcription. Figure 3.75, the CTD facilitates capping and splicing by recruitment of RNA processing factors. A capping enzyme CD is recruited to the vicinity of nascent mRNA by the CTD phosphorylated on SR5. B during transcription, the CTD is phosphorylated on SR2, while the SR5P is dephosphorylated and is involved in recruiting the indicated splicing factors, which define splice sites and facilitates assembly of the spliceosome. In this and subsequent figures, green spheres above the CTD represent relevant CTD binding proteins, while assembled functional complexes are indicated below. Figure 3.76, the CTD functions in 3' processing of both polydenylated and non-polydenylated RNAs. A at 3' ends of polydenylated mRNA, SR2P serves to recruit PCF11, a component of CFII. Human nomenclature is used for all factors. Other 3' end factors, such as CSTF15 and AAUAA binding factor CPSF160 YHH1 in yeast also bind the CTD, whereas SSU72, with the aid of PIN1S1 must dephosphorylate SR5P. The AAUAA element and GU elements are bound by CPSF and CSTF, respectively. Loading of some factors, including CPSF and CSTF, may occur upstream, perhaps at the promoter. B, the 3' end of histone-free mRNA contains a stem loop motif bound by SLBP and a downstream element recognized by U7S and RNP. A complex containing CPSF73, CPSF100, and simplex is recruited for 3' cleavages. THR4P facilitates this process, likely through a yet to be identified factor. C, the 3' end of snRNA genes contains a 3' box that interacts with the integrator complex. RPAP2 binds to SR7P on the CTD and recruits the integrator, and INT11, an integrator subunit, cleaves the RNA. Transport of RNA from the nucleus to the cytoplasm. The transport of RNA to the cytoplasm is central to gene expression. The export of various RNA types that are synthesized in the nucleus is mediated through the nuclear pore complexes by portable export receptors. Small RNAs, i.e., tRNAs and mRNAs, are easily transported by precisely binding to the export receptors. However, large RNAs, rRNAs, mRNAs, and ln assemble into intricate RNP complexes and recruit the exporters through specific adapter proteins. In eukaryotes, the transcription elongation complex is coupled with RNA splicing machinery and a nuclear export complex called TREX, transcription export. This coupling remarkably enhances the fidelity, rate of RNA processing and transfer of mRNA from the nucleus to the cytoplasm. The TREX is formed of heteropentheneric complex, which consists of O2, HBR1, MFT1, THP2, and TEX1, mRNA export factor sub2 and UR1, and SR proteins GBP2 and HRB. During transcription, TREX moves alongside the transcribed gene with RNAPI and binds to the synthesized mRNA transcript and thus feeding it into the export pathway via factor sub2 and UR1. Figure 3.77. Following this, UR1 attaches to the exporter MEX67MTR2, which then binds right to the mRNA as well as to the nuclear pore proteins leading to the transport of mRNP through the nuclear pore complex to the cytoplasm. Figure 3.77, export of RNA from the nucleus to the cytoplasm. The transport is coordinated by multiple proteins that interact with the synthesized mRNA and components of the nuclear pore complex. Nuclear pore complex. Nuclear pore complex, or NPC, is one of the biggest protein complexes in a cell that can be seen by electron microscopy. NPC is a symmetrical structure that resembles a basket extending into the nucleoplasm known as a nuclear basket, and the filaments that protrude into the cytoplasm are called cytoplasmic filaments. Despite a massive size, the NPC is formed of only 30 highly conserved proteins called NUFs that are assembled into biochemically defined subcomplexes. The regulatory activity of NPC is carried out by a subset of NUFs called FG NUFs. FG NUFs characteristically encompass a structure domain named as the anchor point and stem filaments and hydrophilic extensions comprising multiple hydrophobic phenylalanine splicing FG repeats. In humans, the NUF62 complex consists of NUF62, NUF58, and NUF54, each of which contains FG repeats and occupies a central core. NUF62 is also found in the human NUF214 subcomplex housed on the cytoplasmic face of NPC, where it interacts with NUF88 and the FG containing NUF214. Likewise, two other essential subcomplexes, the Y complex and NUF93, form the structural scaffold for NPC and operate as adapters that connect FG NUFs to the nuclear membrane. In humans, the Y complex is composed of 10 proteins NUF37, NUF43, NUF85, NUF96, NUF107, NUF160, NUF133. Sector 13. Say one element in the NUF93 complex consists of NUF35, NUF93, NUF155, NUF188, and NUF205. Human diseases that occur due to faulty RNA processing. The splicing and nuclear export of mRNA are crucial steps for gene expression. A large body of recent studies has highlighted that the RNA processing factors, the core RNA splicing, and export machinery exhibit important regulatory potential. Improper RNA processing is now well established to be the primary cause of several human diseases, especially tumorigenesis and neurological disorders. Mutations in core components of spliceosomes have been documented in several cancers, neurological disorders, and slow onset genetic diseases such as retinitis pigmentosa. Tab 3.8. Structural alterations or mutations can also disturb the folding or expression of spliceosomal components, resulting in a decreased ability of the cell to compensate during stress or aging. Cancer tissues show high enrichment for single nucleotide variations, which contribute to the retention of the intrin and known tumor suppressor genes and therefore end up synthesizing the wrong protein. In the disease beta thalassemia, AG to a nucleotide substitution, an intron one of the concerned gene generates an alternative splice acceptor site leading to retention of a part of the intronic sequence in the mature mRNA transcript. These patients transcribe an abnormally processed RNA species that comprises 19 nucleotides from the 3' end of the retained intron. However, certain variations can be beneficial, e.g., a splice site variant of LDLR that reduces LDL cholesterol in the body and protects against coronary artery disease. Besides splice site mutations, structural modifications that disrupt silencer or enhancer elements are also a common group of disease-causing alterations that impact alternative splicing. Genes diseases mutation are altered. Expressions. Core spliceosome components. PRPF40B myelodysplasia mutation. Acute myeloid leukemia PRPF6 colon cancer amplification. Retinitis pigmentosa mutation PRPF8 acute myeloid leukemia mutation. Retinitis pigmentosa PRP31 retinitis pigmentosa mutation PRPF3. Splicing factors. SF3B1 myelodysplasia mutation U2AF35 U2AF1 chronic myelomonocytic leukemia recurrent mutation SF3A1 acute myeloid leukemia increased expression CRSR2 U2AF65 SF1 SRSF2 SC35 SRSF5 SIRT4 RNA polymerase and its associated transcription factors represent excellent targets for broad-spectrum antibacterial and other classes of drugs as they are highly conserved across the bacterial domain. There are various drugs available in the market having unique transcription targets. The drugs targeting transcription are favored due to various reasons like 1. Bacterial RNAP and its associated transcription factors permit the potential development of broad-spectrum antitranscriptional antibiotics. 2. Also, eukaryotic RNAP is not similar to its bacterial homologue at the sequence level, which suggests low potential cytotoxicity. 3. Numerous high-resolution structures are available, enabling structure-based drug design. Examples of some such drugs are 1. The rifamycin series and fidaxomicin lupiarsen are two series of antibiotics targeting bacterial RNAP available in the market. Rifamycins were the first group of antimicrobials targeting RNAP. 2. Rifamycin SV was the first drug of this class that was used either intravenously or topically. 3. Rifampin RIF synthesized from rifamycin SV is used orally and has broad-spectrum activity against gram-positive, gram-negative, and especially mycobacterial pathogens. 4. Benzoxazine rifamycins have more affinity to RIF-resistant and tuberculosis strains. 5. Streptolide STL acts by inhibiting bacterial transcription initiation and elongation. 6. Saliniamides A and B inhibit transcription in both gram-positive and gram-negative organisms. 7. CBR 703, together with CBR 9379 and CBR 9393, inhibit transcription elongation by stabilizing elongation, complex isomerization, and slow translocation. 8. Microcin J25 is active against gram-negative bacteria and inhibits abortive initiation and elongation by competitively preventing NTP uptake or binding. 9. Estradiol is also a transcription modulating drug with cardioprotective properties. 10. Raloxifene is a transcription modulating drug with improved clinical specificity over first-generation estrogen receptor ligands. In linkage studies in humans and in spontaneously hypertensive rats, it has been found that QTLs influence blood pressure, lipid metabolism, insulin action, and susceptibility to hypertension-induced vascular injury clusters within certain chromosome regions. Certainly, if individual genes are identified that exert pleiotropic effects on all these traits, the controlled expression of such genes might be useful in managing hypertension. 11. Thiazolidinediones drugs exert their effects on all the above causes of hypertension. Troglitazone is a thiazolidinedione approved for the treatment of type 2 diabetes. It also reduces circulating lipids and fatty acids and decreases blood pressure. 12. ANG2 induce changes in gene expression by binding to an angiotensin receptor and a variety of intracellular kinases. Gene regulation in prokaryotes. In a cell, genes are not always constantly expressed. They are turned on or off to respond to the changing environmental conditions and cellular needs. There are multiple factors and processes that control this regulation, such as modification of the chromatin, active to repress and vice versa, regulation of mRNA stability, availability of necessary transcription factors and proteins, etc. The most common and simplest form of gene regulation is the lac operon and TRP operon in bacteria. Lac operon. The bacterium E. coli can utilize either glucose or other sugars like lactose as their source of energy. When grown in a glucose-containing medium, the activity of the genes that metabolize lactose is restricted, but it is turned up significantly when no glucose is available, and bacteria rely on lactose for energy. To uptake and metabolize lactose, E. coli must express a set of three genes latch, LACY, and lack controlled by a single promoter, lac operon, figure 3.78. These genes are transcribed as one RNA. Latch gene encodes the enzyme beta-galactosidase that breaks down lactose into readily usable glucose into lactose residues. LACY gene encodes lactose permes, i.e., a membrane protein which facilitates lactose uptake by the cell. LACA gene translates to a translocase enzyme that is established to transfer an acetyl group from acetyl-CoA to beta-galactosidase. There are two distinct regulators that turn the operon on and off in response to lactose and glucose levels: the LAC repressor and catalytic activator protein CAP. In addition to the three structural genes, the LAC operon also harbors certain regulatory DNA elements which are acted upon by the regulators to control transcription of the promoter, i.e., where the RNA polymerase binds, the operator or repressor site bound by the LAC repressor, and the CAP binding site and activator site that is bound by CAP figure 3.78. The operator overlays with the promoter, and when the LAC repressor protein is stopped, RNA polymerase is unable to bind to the promoter and start transcription. However, the binding of CAP to its site encourages transcription by mediating RNA polymerase to bind to the promoter sequence. Figure 3.78 structure of E. coli LAC operon. It houses three structural genes latch, YN, and two regulatory elements, CAP site and operator that turn the operon on or off for transcription under different circumstances. The LAC repressor serves as a lactose sensor. Under normal scenarios, i.e., when lactose is absent, the repressor impedes transcription of the operon. However, when lactose is present, it promotes transcription of the operon figure 3.79. Apart from recognizing the operator element by its DNA binding site, the LAC repressor also possesses an allosteric site that binds the lactose or its analogs. On binding lactose or its analogs, the repressor protein encounters a change in conformation, leading to an alteration in its DNA binding site. This reduces the binding affinity of the repressor of the operator, and it falls off. This process is termed induction. Thus, the lactose acts as an inducer, and the LAC operon is an inducible operon. However, if both glucose and lactose are present, the cell chooses the best sugar to metabolize first, i.e., glucose. The beta-galactosidase is not synthesized until all the glucose is used. A catalyte of glucose prevents the induction of LAC operon by lactose catabolic repression. This glucose catalyte moderates the level of a key cellular component, cyclic adenosine monophosphate CAMP. The concentration of CAMP is inversely proportional to the glucose levels. During high concentrations of glucose, the cell's CAMP levels remain low and increase correspondingly with a decrease in the concentration of glucose. An increased CAMP concentration activates the LAC operon. The CAMP binds to a protein encoded by CRP gene, CAP to bind to the CAP site. Thus, CAP senses then glucose indirectly. The DNA-bound CAP physically interacts with RNA polymerase to increase transcription of the operon, but only when glucose levels are low. Figure 3.79 operation of the LAC operon in bacteria depending upon the availability of lactose in the external environment. Absence of lactose blocks the operon, while its presence allows transcription at a slow rate. CAP activates transcription in the presence of glucose. Most of the pioneering work to understand the genetic control of LAC operon was carried out by Francois Jacob and Jacques Monod in the 1960s. They studied the mechanisms that regulate the production of beta-galactosidase and lactose permes in the mutants with abnormal beta-galactosidase synthesis. To assess beta-galactosidase, a colorless analog of lactose X-gal 5-bromophore-chloro-3-indyl beta-degalactoside was used as a substrate in a colorimetric experiment. Hydrolysis of X-gal by beta-galactosidase results in the formation of a blue compound. The wild-type bacteria, when grown in the media containing X-gal and lactose, generated blue colonies but produced white colonies when grown in the media containing X-gal and glucose. In the latter case, cells are unable to hydrolyze X-gal due to a deficiency of beta-galactosidase yielding white colonies. However, on being exposed to chemical mutagens, E. coli cells generated some blue colonies when plated on X-gal glucose plates. These cells, when further grown in glucose-containing media, were observed to express all the genes of the operon at a significantly higher rate than their neighboring wild-type cells. Such cells, referred to as the constitutive mutants, fail to suppress the LAC operon during lack of lactose but instead show continuous expression. Next, they tested whether this failure of the repression is due to the inability of the repressor protein to bind to the operator element or due to a faulty repressor protein itself. They studied the mutations in the operator element and the repressor. Jacob and Monod mutagenized E. coli cells that carried a copy of the wild-type repressor gene I+1 each on the bacterial chromosome and a plasmid. To form a repressor constitutive mutant, I isolated mutations in both copies of the repressor gene must occur, which was less probable. On the contrary, a single mutation in the operator could yield a constitutive mutant OC. In this manner, they determined the mutants that constantly expressed the LAC operon despite the presence of two copies of the wild-type repressor gene. Further analysis of the effect of different mutations on the activity of LAC operon provided valuable insights into the process of genetic control of gene expression. In these experiments, the activities of beta-galactosidase and permes were checked in the presence and absence of an inducer molecule isopropyl beta-deogalactoside IPTG. The findings revealed that OC mutation is dominant over the wild-type operator sequence O+, and it acts in cis, i.e., it only affects the expression of the genes present on the same DNA molecule. These OC mutations do not allow the binding of the repressor protein to the operator element. Unlike dominant OC, I is recessive in nature with respect to I+, and it was observed that I+ could regulate the genes on different DNA molecules, i.e., act trans due to its ability to encode a protein repressor that can freely diffuse in a cell. Genes. Sequence product. Sequence types functions important mutants. Protein producing genes. Lacey repressor protein. Contains two binding sites, one for the operator and one for allolactose, the inducer I, unable to bind to the operator IS, unable to bind the inducer allolactose. Latch B-galactosidase cleaves lactose into two monosaccharides, glucose and galactose. C. No functional B-galactosidase. Lacey permease facilitates lactose transport across the cell membrane. Y. No functional permease. Lactotransacetylase protects against harmful byproducts of lactose. Metabolism. A. Number transacetylase. Genes. Sequence product. Sequence types functions important mutants. Regulatory sequences. LACO operator binds repressor protein to block transcription of operon. Genes. OC fails to bind. Repressor protein. Lacey P promoter binds RNA polymerase P, fails to bind RNA polymerase or so. Weekly. Table 3.9 lac operon genes and regulatory sequences. Regulatory mutation in lac operon. In the 1950s, Jacob, Monod and Hardy identified rare mutant strains of bacteria which had abnormal lactose metabolism. One type of mutant designated Lacey resulted in the constitutive expression of the lac operon. Even when the lactose was absent, lac operon in these mutants continued to express. The exact mode of action of the lac repressor was not known at that time. It was thought to produce an activator for the operon, which kept it transcriptionally active throughout. To understand the nature of this mutation, they applied a genetic approach in 1961. It involved bacterial mating by conjugation. The circular segments of DNA are called F factors. Sometimes, the F factors carry the genes that were originally present in the chromosome. Then they are called F factors. They identified F factors which carry the Lacey gene and lac operon. These F factors can get transferred from one cell to another by conjugation. The strain of bacteria containing F factors is called merozygote or partial diploid. Mutations in regulator gene. Lac repressor encoded by the Lacey gene has two binding sites, one for binding the DNA at the operator site and the other for binding allolactose. There are Lacey mutants that either fails to produce lac repressor or the repressor is unable to bind to the operator site. Hence, the lac operon remains constitutively active both in the presence and absence of lactose. There are other regulatory mutations known for the Lacey gene. Lacey S mutation produces a repressor that cannot bind allolactose. Hence, it always remains bound to the operator tab 3.10. Even if lactose is present, the operon cannot be induced, creating a super repressed state. Genotypes. BB-galactosidase permease. Descriptions. Lactose number. Lactose number. Lactose. 1. I, P plus O plus C plus Y plus. Constitutive transcription due to LACO mutation. 2. I plus P plus O C Z plus Y plus. Constitutive transcription due to LACO C mutation. Genotypes. BB-galactosidase permease. Descriptions. Lactose number. Lactose number. Lactose. 3. ISP plus O plus Z plus Y plus. Transcription is not inducible due to LACO-S. Mutation. 4. I plus P, O plus Z plus Y plus. No effective transcription due to Lacey B mutation. Table 3.10 synthesis of beta-galactosidase and permease by haploids and partial diploids with regulatory gene mutation. Mutation in structural genes. Mutant strains which had lost the ability to synthesize beta-galactosidase or permease were identified in Aptolach and Lacey structural genes, respectively. These mutations led to changes in the amino acid sequence and structure of these proteins which cause loss of function in most cases. From the study of partial diploids, they could infer that Latch and Lacey mutations are independent of each other. Partial diploids with Latch plus Lacey on plasmid and Latch Lacey plus on bacterial chromosome produce normal enzymes tab 3.11. Hence, they could conclude that a single functional gene, either in the chromosome or in the plasmid, is capable of producing a normal lac operon phenotype. Genotypes. BB-galactosidase of permease. Descriptions. Lactose number. Lactose number. A symbols plus N indicate production and no production, respectively, of functional enzymes. Table 3.11 synthesis of beta-galactosidase and permease by haploids and partial diploids with structural gene mutation. Mutation at DNA binding site. Jacob and Monod could identify mutants having defective promoter regions. Those were called Lacey P mutations. RNA polymerase is not able to bind to the defective promoter. These mutant strains do not produce any protein of the lac operon either in the absence or presence of lactose. Lacey P mutations are cis-acting and hence inhibit the synthesis of genes present in the same DNA molecule. Therefore, Lacey plus Lacey P, Latch plus Lacey plus Lacey P plus Latch will not produce functional B-galactosidase. Mutations in the operator site. They identified certain mutations where B-galactosidase was produced even in the absence of lactose. The mutation was mapped to the operator region. It was called a constitutive operator or LACO-C. Such mutation does not allow the binding of the repressor. Hence, RNA polymerase can constitutively bind to the promoter, and transcription continues even in the absence of lactose. A partial diploid Lacey plus LACO-C Latch plus Lacey plus LACO plus Latch plus shows constitutive production of B-galactosidase. It showed that constitutive mutation LACO-C is dominant over LACO plus. In Lacey plus LACO plus Latch plus Lacey plus LACO-C Latch did not show constitutive B-galactosidase production. Therefore, the operator mutations affect genes which are present on the same molecules. TRP operon. The tryptophan operon in E. coli encodes necessary enzymes for the amino acid. Tryptophan is turned on or off, respectively, in response to low or high levels of tryptophan. The TRP operon consists of 5 genes TRPA, TRPB, TRPC, TRPD, and TRPE that code for enzymes necessary for tryptophan biosynthesis, a promoter for binding of RNA polymerase and an operator for binding of a repressor protein figure 3.80. The TRP operon is a repressible promoter, i.e., the tryptophan acts as a repressor. Like lac operon, the regulatory protein TAR repressor binds to the operator and preserves the operon from getting transcribed by physically blocking the binding of RNA polymerase. In the presence of tryptophan, repressor proteins undergo structural change and are activated. The activated repressor protein binds to the operator and restricts transcription only in the presence of tryptophan. On the contrary, when there is less availability of tryptophan in the cell, the TRP repressor remains inactive and does not block transcription, thus allowing the operon to be transcribed by RNA polymerase figure 3.80. Thus, tryptophan behaves as a self-governor by deciding its own synthesis through a negative feedback mechanism. Figure 3.80 operation of TRP operon in E. coli. Presence of tryptophan inhibits RNA synthesis by the operon, whereas the absence of tryptophan facilitates transcription. Besides directly sensing the tryptophan levels, there is a second layer of negative regulation known as attenuation that also persists. The attenuation dampens the TRP operon and was first recognized from the effects of mutations in the TRP repressor gene. Mutations that disrupt the function of the TRP repressor protein potentially interrupt the negative feedback loop. Such mutants continue to synthesize tryptophan even when it is present in the environment. Surprisingly, these repressor mutants, when grown in tryptophan deficient conditions, produce even more tryptophan than the wild-type bacteria which have been starved for tryptophan. This suggests that there is an additional regulatory mechanism besides the repressor protein that senses tryptophan availability. This attenuation of TRP operon is governed by a regulatory element called the leader, present within the transcribed mRNA, upstream of all codons of TRP structural genes. The leader causes premature termination at the attenuation locus present between the operator site and codons of structural genes, resulting in inhibition of operon expression. The leader sequence can attain different stable conformations, amongst which only one conformation permits the translation of the remaining operon. In order to sense tryptophan levels, the leader sequence encodes a small peptide that harbors TRP codons. When TRP is abundantly available, this leader peptide is easily translated and causes mRNA-based pairing to prevent the transcription and translation of operon. On the other hand, when the environment is deficient in tryptophan, there is no translation of the leader peptide, and the leader assumes a second conformation that allows the transcription and translation of the operon. Thus, the structure of the leader sequence serves an important role in mediating attenuation. Modulation, transcription, attenuation. While all the above mentioned control mechanisms regulate gene expression at the transcription initiation steps, a second mechanism known as transcriptional attenuation regulates gene expression at the elongation and termination steps by interfering with the ability of RNA polymerase to continue elongation past specific sites. In addition to regulation at the transcription initiation level, the TRP operon is also regulated by transcription attenuation, which provides an added level of control, resulting in much tighter regulation than could be achieved by repression of initiation alone. Mechanism of attenuation. A162 base segment, also known as the leader segment, is located between the 5' end of the TRP mRNA and the start codon of the TRPL gene. After transcription initiation, most mRNA molecules are terminated in the leader region except in tryptophan deficient conditions, yielding a 40 nucleotide long, short RNA molecule which terminates before the structural genes of the operon are transcribed. This region in which termination occurs is a regulatory region called the attenuator. Transcription termination is signaled by the usual stem loop structure followed by a sequence of 8 astronomical unit pairs. The mechanism of attenuation depends on the fact that in bacteria, translation and transcription are coupled with each other such that ribosomes begin translating the 5' ends of an mRNA while transcription is still going on. Thus, the translation rate can impact the structure of the grown RNA chain and determine the fate of further transcription. When tryptophan is present, translation proceeds at a normal rate, as a result of which the stem loop terminator structure is formed, and mRNA synthesis is halted. However, in tryptophan deficient conditions, protein synthesis is stalled, and the ribosomes bound to the mRNA prevent the formation of the stem loop terminator sequence, thus allowing mRNA synthesis to continue. The leader sequence plays a vital role in regulating gene expression. It has two adjacent tryptophan codons at positions 10 and 11, which affect the rate of translation. These codons provide a link between transcriptional attenuation and tryptophan availability such that in tryptophan deficient conditions, the ribosome stalls at this point and transcription of TRP mRNA continues. However, when tryptophan is available, the translation continues while transcription is terminated. Figure 3.81. As illustrated in figure 3.81, the transcription and translation of the TRP mRNA are coupled and occur simultaneously. In tryptophan adequate conditions, the ribosomes trail only slightly behind the growing mRNA chain such that while the ribosome is bound to segment 2 of the mRNA, segments 3 and 4 hybridize with one another to form a stem loop structure that signals transcription termination. Under tryptophan depleted conditions, the ribosomes stall at region 1 of the mRNA, which contains the two tryptophan codons. Due to the stalling, the ribosomes trail far behind the growing mRNA chain, and segment 2 remains exposed and unbound. The unbound segment 2 is now free to form an alternate stem loop structure with segment 3. This prevents the formation of the 3 to 4 stem loop structure. Transcription is thus not halted and can now proceed way beyond the attenuator sequence. Figure 3.81 regulation of TRP operon by attenuation. A transcription and translation of the TRP mRNA occur simultaneously. When tryptophan is available, the ribosomes trail only slightly behind the growing mRNA chain. When the ribosome interacts with segment 2 of the mRNA, segments 3 and 4 of the growing mRNA transcript are free to hybridize with one each other and form a stem loop structure which signals transcription termination. When tryptophan is present either at low levels or is completely absent, the ribosomes trail far behind the RNA polymerase due to ribosome stalling of the two tryptophan codons which are present in segment 1 of the mRNA. Segment 2 of the mRNA is thus left unexposed, and it is free to hybridize with segment 3. The segment 2 to 3 interaction prevents the formation of the 3 to 4 terminator loop, and transcription can thus continue. Gene regulation in eukaryotes. There are no operons in eukaryotes, and apparently, the regulation of gene expression is much more complex. Like bacteria, different proteins bind to the regulatory elements to govern transcription by altering RNA polymerase activity. Besides proteins, non-coding RNAs, chiefly learners and elastinase are key players of genome regulation. Packaging of eukaryotic DNA into dense and loose chromatin along with its wide-ranging modification adds another layer of regulation in gene expression. Gene regulatory elements. As described in section D2.6, besides core promoter elements, Tata box and INR, cis-regulator elements, CAAT box, GC box, and DPE region serves important seats for binding of general transcriptional factors and other essential proteins. In addition, distal regulatory elements, enhancers, and silencers exert their effect on transcription by interacting with transcriptional machinery achieved by chromatin looping. All these elements are bound by specific transcriptional regulatory proteins. Tab 3.12. Structure was first determined by studying Gal4 protein in yeast through successive experiments. It was noticed that this TF is composed of two separate functional domains, one a DNA binding domain at the N-terminal end to interact with specific DNA elements, and two an activation domain at the C-terminal end to interact with other proteins to increase transcription at a nearby promoter. Another yeast TF GCN4, a TF that regulates the genes necessary for amino acid synthesis, was also found to possess a 60 amino acid DNA binding domain at its C-terminus and a 20 amino acid activation domain near the middle. Today, the structure of several transcription activators has been studied in detail. The structural model of eukaryotic activators has revealed that these TFs contain one or more activation domains which are connected through relatively flexible protein domains to a sequence-specific DNA binding domain. Activation domains operate via protein-protein interactions with the bound TFs at the promoter. The flexible protein domains control the spacing between the two functional domains, activation and binding, to regulate the interaction of these domains with TFs at the promoter. In such a scenario, if the DNA binding domains of adjacent TFs get reallocated from their original positions on the DNA, the activation domains can get interact due to the presence of these flexible protein linkers. Types of DNA binding domains. TFs are largely classified depending upon the type of DNA binding domain they possess. Eukaryotic TFs comprise various structural motifs that bind to definite DNA elements. A large body of structural motifs presents alpha helix to the major groove of DNA, wherein the protein atoms of TF feature particular hydrogen bonds and van der Waals interactions with the atoms of DNA. Nearly all structural types of DNA binding domains underline a characteristic consensus amino acid sequence. Common classes of DNA binding domains have been discussed here. Transcription factors. Consensus DNA binding sites. Specificity protein 1 SP1 GGGCGG. CCAAT enhancer binding protein CEBP. CCAAT. Activator protein 1 AP1 TGACA. Optimer binding proteins OCT1 and October 2. ATGCAAT. EBOX binding proteins E12, E47, E2 to 2. CANNTG. Table 3.12 transcriptional regulatory proteins and their binding sites on the DNA. Transcription activators. Transcription activators are modular proteins that constitute distinct functional domains and bind to distal regulatory regions of the gene. There. Homeodomain HOX TFs. Members of this family e.g., CDX2, HOXA1, NAN. Members of this family e.g., CDX2, HOXA1, NANOG, October 1, POU3F2, TGIF1, ZEB1 feature a homeodomain composed of 60 amino acid helix turn helix DNA binding domain. The DNA region that encodes a homeodomain is referred to as a homeobox, and homeobox containing genes are called HOX genes. A homeodomain is a very conserved motif in figure 3.82 a homeodomain and its interaction with the DNA. Contains 3 helical regions that fold into a tight globular structure enclosing a 5'TAAT3, core motif figure 3.82. The recognition helix within the homeodomain binds to the major groove of DNA while the N-terminal tail joins with the minor groove. Homeodomain is essential for shuttling of HOX TFs between the nucleus and cytoplasm. HOX TFs play a major role in patterning of the anterior-posterior axis in embryo and organ development. It is a helix-turn-helix motif containing 3 alpha helices connected by loops of amino acids. Zinc finger proteins. Numerous proteins like KLF4, ZNF281, BRCA1, BDR, GATA4, MAP3K1, and MORC1 fold about a central ZN2 plus ion to form a compact domain termed as zinc finger that coordinates cysteine and histidine residues in different combinations. Zinc finger proteins ZNFs are the most abundant group of proteins that are implicated in several cellular processes, namely regulation of transcription, ubiquitin facilitated protein degradation, signal transduction, DNA repair, and cell migration. Transcription factor IIIA tibia from Xenophus laevis was the first CNF that was studied. A classical zinc finger motif C2H2 consists of 2 cysteines in one chain and 2 histidines in the other and is coordinated by ZN2 plus between 2 antiparallel beta sheets and 1 alpha helix figure 3.83. Fork head box Fox proteins. Fox family of TFs has been named after the fork head mutants of Drosophila. Melanogaster comprises a winged helix turn helix DNA binding domain. The fork head motif comprises 3 N-terminal alpha helices, 3 beta strands and 2 loops towards the C-terminal that are folded peculiarly to resemble the wings of a butterfly. Therefore, the name winged helix. The TFs FOXP1, FOXO3, FOX1, FOXO1, and linker histones H1 and H5 are the members of this family of TFs. Fork head factors serve as terminal effectors of multiple important signal transduction pathways such as the TGF-beta, the MAP kinase pathway, and the insulin growth factor pathway. Figure 3.83 classical C2H2 zinc finger motif. The C2H2 zinc finger motif comprises a short antiparallel beta sheet shaped by 2 strands and hairpin turn and is followed by an alpha helix. Figure 3.84 fork head motif. It comprises 3 of each alpha helices and beta sheets and 2 loop regions. Leucine zipper proteins. The first TFs that were identified in this category possess the hydrophobic amino acid leucine at every 7th position in the C-terminal region of their DNA binding domains. These TFs bind to DNA as dimers, and the repetitive leucine residues are crucial for dimerization. The dimeric protein consists of 2 extended alpha helices that hold the DNA at 2 successive major grooves like a pair of scissors, separated by nearly half a turn of the double helix figure 3.85. This family comprises TFs like activator protein 1, AP1, FOSGEN, and CCAAT enhancer binding protein beta, CEBP beta. Figure 3.85 basic leucine superdomain in AP1. It comprises numeric protein with leucine residue at every 7th base in the DNA binding domains. Helix loop helix proteins. The DNA binding domain of this class of TFs is like the zipper motif, but a non-helical loop of the polypeptide separates two alpha helical regions in each monomer. This motif contains an internal alpha helix with basic amino acid residues to interact with DNA, a middle loop region, and AC terminal region with hydrophobic amino acids set apart at intervals that are typical of an amphiphasic alpha helix. Figure 3.86 helix loop helix proteins. A the HLH domain. B the predicted 3D structure of the full-length protein. Transcriptional repressors. Repressors primarily function by either blocking the binding of activators to regulatory elements or by directly interacting with the general TFs to inhibit transcription. Unlike transcription activators, several eukaryotic repressors also possess two functional domains, i.e., a denabbing domain and a repression domain. Most of the amino acid regions that serve as repression domains are comparatively shorter, less than 20 amino acids, and have a high number of hydrophobic residues. Repression domains can also be sometimes larger and well-structured domains. The ligand binding domain RXR alpha behaves as a repression domain during the lack of ligand but as an activation domain when it binds to its cognate ligand 9-cis-retinoic acid. Chromatin structure and modifications. Chromatin structure is a primary factor that decides a gene's expression. As previously discussed in section D2.6, eukaryotic DNA is tightly wrapped around the nucleosome pores to form the chromatin and needs to be free to begin transcription. This packaging of DNA not only restricts the activity of RNA-P2 but also inhibits the binding of essential TFs. This constraint of nucleosomes is eased by chemical modification of specific residues of histone proteins or DNA sequence and or binding of certain remodeling proteins along with the genes that must be actively transcribed. Histone modifications. The histone proteins are highly basic in nature, and their N-terminal tails hang out from their nucleosome pore in order to interact with the neighboring nucleosomes. Chemical modification like acetylation, methylation, ubiquitination, phosphorylation, etc. of certain residues of these tails can drastically affect the overall chromatin structure. Acetylation of the lysine residues is highly dynamic and is mediated by the activity of two enzymes histone acetyltransferases, HATs, and histone deacetylases, HDACs. The HATs neutralize the positive charge of lysine residues by attaching the acyl group to the epsilon amino group of lysine side chains. This neutralization weakens the attachment between histones and DNA, thereby releasing the nucleosomes at the modified site. This frees the chromatin, and the underlying gene is successfully transcribed. On the contrary, HDACs are chiefly transcriptional repressors. HDACs deacetylate lysine residues, restoring their positive charge to stabilize the local chromatin. The histone mark H3K27AC red is acetylation of 27th lysine residue K of H3 histone protein is the most common mark that is enriched with the promoter and enhancer elements of actively transcribed genes. Unlike acetylation, methylation does not change the charge on the histone protein, and it mostly takes place on the side chains of lysine and arginine residues which can be mono, di or triangulated. The enzymes methyltransferases catalyze the transfer of a methyl group from S-adenosylmethionine SAM to lysine epsilon amino group or an arginene's omega guanidino group. The activity of methyltransferases is upturned by another enzyme, demethylases that act to get rid of methyl groups from lysine and arginine residues. The histone marks H3K43 and H3K41 read respectively as trimethylation and monomethylation of fourth lysine residue of H3 histone protein individually represent an active promoter and enhancer regions of the gene, but the histone signature H3K27E3 denotes an active chromatin. Acetylation and methylation end up in small changes in amino acid side chains. However, the process of ubiquitination adds a much larger molecule ubiquitin, 76 amino acid long, to the lysine residues of histone proteins via the consecutive activity of three enzymes E1 activating, E2 conjugating, and E3 ligating enzymes. The two well-known sites that undergo ubiquitination reside within histones H2A and H2B. Monoubiquitination of lysine at position 119 of H2A, H2AK119 is important for gene silencing, whereas H2BK123 functions in the initiation and elongation phases of transcription. Phosphorylation of histones predominantly happens on serines, threonines, and tyrosines in the N-terminal tails and is mediated by kinases and phosphatases that respectively incorporate and erase the modification. Like acetylation, histone phosphorylation is also a highly dynamic process. DNA methylation. Besides histones, methylation of the chromatin is another highly prevalent mechanism that determines gene expression in vertebrates. Certain cytosines that precede guanosine residues, CpG sites, in the DNA are modified by the addition of the methyl group at the fifth carbon position. DNA methylation is associated with decreased transcriptional activity, especially for the genes that harbor a large number of CpG sites near the promoters. Transcriptional repression is mediated by the action of the protein MECP2, which particularly binds to the methylated DNA. Remodeling proteins. Specific proteins called nucleosome remodeling factors modify the structure of nucleosomes, nucleosome repositioning, to increase the accessibility of the wrapped DNA for the binding of essential TFs. Apart from these, the constraint of the nucleosomes is relieved by the binding of high mobility group proteins, HMG14 and HMG17 that alter the interaction of H1 linker histone with nucleosome pore octamers to maintain open chromatin, thereby allowing transcription to initiate. Regulation by non-coding RNAs. Though not translated, it is now well established that non-coding RNAs play a crucial role in the regulation of gene expression. The most common non-coding RNA species that are extensively characterized for gene regulation in eukaryotes are elincinase, murines, and recently, cercinase. Long non-coding RNAs. The number of LNCRNA genes is nearly equal to the protein coding genes. Most of the elincinase are synthesized and processed in a similar process of protein mRNAs. Today, elincinase are the largest class of non-coding RNAs that determine gene expression and have immense regulatory potential starting from transcription catalysis and chromatin remodeling to RNA-mediated silencing of entire chromosomes. Figure 3.87. Elincinase perform a wide range of functions starting from organization of genomic DNA into speckle bodies, histone modifications and direct regulation of transcription in the nucleus to regulating mRNA stability, protein translation in the cytoplasm. Several elincinase have been documented to function in cis or trans to modulate protein gene expression through altered chromatin context. Elincinase, hot air, COX transcript antisense RNA, and ANRIL CDKN2B antisense RNA1 physically associate with an active scaffold for concerted actions of chromatin modifying complex, polycomb repressive complex PRC to deposit repressive histone mark H3K27E3 at the targeted loci. However, a yeast LNCRNA SRG1 represses gene expression by directing a higher number of nucleosomes to the promoter of phosphoglycerate dehydrogenase gene SCR3. A direct impairment of gene transcription by LNCRNA has been observed for DHFR, dihydrofolate reductase locus, wherein the formation of LNCRNA DNA triplex structure inhibits the assembly of the transcription pre-initiation complex. Additionally, elincinase have been implicated in the upregulation of gene expression via enhancer-mediated complex furnace. A human runner regulates transcription of a highly conserved NADPH-dependent enzyme gene DHRS4 by enhancing the expression of an antisense LNCRNA, DHRS4AS1 through chromosomal looping. Depending upon what proteins are recruited, LNCRNA can act as a co-activator or co-repressor to specific DNA regulatory elements. Besides acting as a scaffold for chromatin modifiers, LNCRNA can mimic DNA. LNCRNA GAS5 growth arrest specific 5 represses glucocorticoid receptor GR gene expression by folding into a structure that mimics the DNA binding site of GR. By contrast, LNCRNA NRON, non-coding repressor of NFAT, promotes a repressive chromatin environment by Figure 3.87 common functions of elincinase. Obstructing the nucleocytoplasmic shelling of the transcription factor NFAT, nuclear factor of activated T cells, via competitive binding to transport receptor important beta. Apart from transcription, elincinase also regulate mRNA processing and stability. Modulation of chromatin histone signatures by highly conserved nuclear antisense LNCRNA within human FGFR2 locus encourages epithelial-specific alternative splicing of FGFR2 mRNA by hindering the binding of a repressive chromatin splicing adapter complex needed for mesenchymal-specific splicing. An exonic antisense LNCRNA BACD1AS increases the stability of base 1 beta site activating enzyme 1 mRNA by forming a stable RNA-RNA duplex and limiting the binding of mRNA 485, leading to diminished repression of the base 1 gene. Apart from competing with mRNAs for binding sites on target mRNAs, elincinase sometimes function as decoys to attenuate mRNA regulation, mRNA sponge. For instance, a muscle-specific LNCRNA LINCMD1 binds and sequesters near 133 and MIR 135 in order to indirectly activate the mRNA-dependent downregulation of key transcription factors MEML1 and MEF2C needed for muscle differentiation. Elincinase are also being acknowledged as translational regulators. A mouse antisense LNCRNA UPL1AS upregulates the translation of UPL1 mRNA, ubiquitin carboxyl terminal esterase L1 through repeat element SINB2 in its 3' non-overlapping region without changing UPL1 mRNA levels. Thus, elincinase are versatile biochemical molecules that function in diverse contexts governing all aspects of gene expression. MicroRNAs. Murines are the dominant class of small non-coding RNAs that serve as guide molecules in the RNA silencing mechanism in a cell. Targeting nearly all protein coding transcripts, murines play an important role during a cell's developmental and pathological process. The biogenesis of murines is tightly controlled. Dysregulation in mRNA synthesis has been established to contribute to different human diseases: type 2 diabetes, cancers, cardiovascular diseases, Alzheimer's disease, schizophrenia, rheumatoid arthritis, polycystic liver disease, psoriasis, etc. All mRNAs are transcribed as primary molecules, which are processed into precursor mRNAs, and finally mature mRNAs of 20 to 22 nucleotides in length, figure 3.88. This processing is facilitated by two RNase-free proteins Roche and Dicer. mRNAs degrade the target mRNA by recruiting the minimal mRNA-induced silencing complex nearest. This complex consists of a guide strand and an argonauts, a GO family of proteins. During RNA silencing, mRNA base pairs with its target mRNA, and the GO protein recruits factors that encourage translational repression or decay of the bound mRNA. The target specificity of nearest is underlined by its interaction with complementary sequences on target mRNA, termed mRNA response elements (MREs). A fully complementary mRNA hybrid induces the endonuclease activity of a GO, leading to the cleavage of the bound mRNA. The binding sites for mRNAs are generally housed in the 3' untranslated regions (UTR) of mRNAs. The nucleotide sequence of the 5' ends of mRNA span in the position 2 to 7 is termed as mRNA seed region and is essential for target recognition. Nearly 60% of the human protein coding genes contain at least one conserved site for mRNA binding. This highlights that the expression of most of the protein genes are strictly regulated by mRNAs. Mature mRNAs mostly interact with the 3' UTR of the target mRNAs, resulting in their degradation to repress translation. However, some mRNAs are reported to bind the 5' UTR encoding sequence of a gene and promoter element. The binding of mRNA to 5' UTR or the coding sequence has been seen to exert silencing of gene expression, while the interaction of mRNA with the promoter is known to induce transcription. Under specific cellular conditions, mRNAs can also induce gene expression. Several mRNAs, such as LET7, activate translation during the cell cycle arrest by interacting with an argonaute protein GO2 and Fragile X mental retardation related protein 1 FXR1. Besides, mRNA-induced upregulation of gene expression has also been reported in quiescent cells such as oocytes. mRNAs have also been revealed to interact with the enhancer elements that transcribe enhancer-derived RNAs to increase the transcription of adjacent genes by fostering transcriptionally active chromatin while modifying the alternative splicing profiles. Nuclear mRNA 522 suppresses the transcription of the 2 times 10 to the 1 cypric counts gene by interacting with the DNA stem loop structure within the promoter of the gene. Figure 3.88 synthesis and action of mRNAs. mRNAs are produced endogenously as large precursors that are spliced to generate a mature mRNA. With the help of argonaute proteins, mRNAs degrade the target mRNA transcript. mRNAs can be released into extracellular fluids and transferred to the target cells through exosomes. Extracellular mRNAs serve as chemical messengers to facilitate cell communication. Today, circulating levels of certain mRNAs are used as biomarkers for assessing human diseases. Circular RNAs. Cercenas are a novel category of non-coding RNAs that have been recently identified and are being extensively studied. Different from the linear RNAs, cercenas are covalently closed single-stranded loop structures created by alternative splicing of a pre-mRNA in which an upstream splice acceptor is linked to a downstream splice donor in a process termed back splitting. Figure 3.89. Figure 3.89 process of formation of cercenas in an isoform diversity. Instead of a canonical splicing event, an alternative splicing event results in the joining of a downstream splice site back to the upstream site. Cercenas can arise endogenously from exons, exon-intron or introns, and are extremely stable and specific. They are expressed in many eukaryotic organisms in a species, tissue, disease, and developmental stage specific manner, and their expression are conserved across mammals. Cercenas have been shown to have multifaceted functions. They are the competing class of endogenous RNAs and can control gene expression by sponging microRNAs, binding necessary proteins or getting translated into a protein. A cercena named circuitable form from a developmentally regulated gene Drosophila musclin MBL tunes the expression of the MBL by competing with the synthesis of mRNA and binding to MBL protein. Another cercena CIRS7 is highly expressed in the brain of mammals during development and is a derivative of the CDR1 antisense locus. It serves as a mRNA sponge featuring more than 70 potential mRNA binding sites for MIR7. Upon induction of ectopic expression of CIRS7 in zebrafish, which does not express CIRS7 endogenously but expresses MIR7, defects in midbrain development are noticed. The findings implicate that this RNA governs the development of the mammalian brain when CIRS7 and MIR7 are co-expressed. Similarly, a testis-specific cercena SRY, sex-determining region Y, is known to sponge and govern the functions of MIR138 in human embryonic kidney HEK293 cells. Besides, endogenous cercenas are demonstrated to be translated into a functional protein. CircFBXW7 translates a 21 kDa protein called FOX and WD repeat domain containing 7FBXW7. An overexpression of FBXW7 results in decreased cellular proliferation and represses glioma tumorigenesis. A few studies have assessed the protein binding capacity of cercenas. Human cercena CDR1 is, i.e., cerebellar degeneration-related protein 1 transcript, have been seen to strongly interact with the proteins which are known to mediate mRNA function. Cercenas have been linked with different human diseases where they are disease protective in some cases and disease inducing in others. Given the disease regulatory mechanisms, cercenas are functional biomarkers and therapeutic targets for different disorders. RNA editing. Modifications in RNA sequence can affect its structure, function, or stability and can potentially change the informational content of the transcribed molecule. RNA editing refers to any site-specific alteration in the sequence of RNA after it is transcribed from the genome, on top of the processing events, splicing, polyadenylation, or capping. Editing can occur in any of the classical RNA types, rRNA, mRNA, or ternal or even non-coding RNAs, and has been recognized in a large variety of organisms, starting from unicellular protozoa to humans. Such sequence revisions comprise nucleotide insertions, deletions, or conversions. For instance, the mitochondrial RNAs in Trypanosoma brucei exhibit wide-ranging insertions and deletions of uracil residues. Plant chloroplasts and mitochondria transcripts undergo extensive cytosine to uracil editing. Adenosine to inosine editing. The change of adenosine-based to inosine ATI by deamination at C6 position, which is carried out by adenosine deaminase enzymes. ADAR is the most common type of RNA editing. Figure 3.90. Figure 3.90. The ATI RNA editing by ADAR proteins ADARs result in a hydrolytic deamination reaction that transforms into I-nucleotide. Since an adenosine base pairs with uridine, following the editing, the altered nucleotide inosine acts as guanosine by base pairing with cytidine. Like so, an ATI RNA editing ends up in the addition of amino acids that are not originally encoded in the genome. Three members of the ADAR family are encoded in mammals: ADAR1, ADAR2, and ADAR3. The enzyme ADAR1 directs most of the RNA editing events, though it is expressed ubiquitously with ADAR2. In contrast, ADAR3 is catalytically inactive and is chiefly expressed in the brain at much lower levels. ADARs specifically bind to double-stranded RNAs, dsRNAs, and feature conserved functional domains. The RNA binding domain of ADAR is nearly 65 amino acids long and exhibits an alpha-beta-beta-beta alpha conformation. Its C-terminal region comprises the deaminase domain that is the catalytic core of ADAR. Then there are other structural elements that are unique to certain enzymes, e.g., ADAR1 possesses 2 zDNA binding domains C-alpha and Z-beta, and ADAR3 has an arginine-rich single-stranded RNA binding domain or the R domain at its N-terminal segment. X-ray crystallographic study of human ADAR2 has elucidated that his 394, Lu 396, CY 451, and C516 in the coordination of the ZN2 plus 4 in the catalytic core and a base flipping positions the selected adenosine are to carry out the deamination. Besides, a nocetyl hexaphosphate or INSP6 is also found inside the enzyme core. The INSP6 is surrounded by multiple arginine and lysine residues and is envisaged as an important molecule for deamination. ADARs identify both intermolecular and intramolecular dsRNAs, which are longer than 20 nucleotides and can edit more than 50% of the existing adenosines for longer transcripts, and GT, 100 bases long. Editing can generate new proteins or alter their function, modify gene regulation, and can even confer immunity against endogenous dsRNAs. Figure 3.91. Many RNA editing events have been reported in the mammalians in the protein coding sequences. Figure 3.91 Functional consequences of RNA editing. Editing within the exonic portion of an RNA can result in the substitution of existing amino acids or lead to the synthesis of a new protein. B editing can also disrupt the present splicing signals or create additional splice sites. C editing in non-coding transcripts can create or destroy the recognition elements of mRNA and the UTR of the RNA. D editing in mRNA sequence can modify the mRNA itself that may recognize a different set of target mRNAs. E editing of the parent sequence can affect or disrupt the processing of cercenas. Cytidine to uridine editing. Besides ATI editing, another common deamination converts cytidine to uridine. This editing occurred at the C4 position of cytidine and was originally found in vertebrates for transcripts that encode apolipoprotein B, a POP, wherein a glutamine codon CAA gets converted into a stop codon UAA. Figure 3.92. This editing involves a tripartite regulatory region around the edited cytidine and a multiprotein complex known as the E-editosome. The three-way regulatory motif that edits the cytidine residue at the position 6666 within the APOP transcript comprises an 11 bases region, UGA, UCA, GUA, UA, referred to as the Morin sequence, a 4 to 6 bases spacer region between the modified cytidine and the Morin sequence, and a regulator element located just upstream of the altered cytidine. The E-editosome consists of a 27 kDa catalytic cytidine deaminase called APOBDC1 and other supplementary proteins. This editing is also observed for a tumor suppressor gene involved in neurofibromatosis type 1 that encodes neurofibromin and several mitochondrial and chloroplast transcripts of higher plants. Figure 3.92 Cytidine and adenosine deaminases are critical RNA editors that play important functions in physiological events. The vital role of APOBDC1 editing can be observed in the production of apolipoprotein B in the gut. The CDU editing at residue 2153 of hepatic ApoB100 transforms the glutamate to a stop codon and produces a truncated protein ApoB48 in intestinal cells 4. B in neurons. mRNA editing of the glutamate receptor 2, LUR2, at position 607 by DAR2 results in adenosine to inosine chance. This transforms the CAG codon for glutamine Q to sig for arginine R as CGG since ribosomes adenosine I as guanosine G. This neutralizes the diffusion of divalent cations and makes the receptor impermeable to calcium. Uridine to cytidine editing. Unlike the deamination, ADI, and CDU, the uridine to cytidine editing involves amination. The U to C editing is not fully understood but is thought to be similar to the catalysis process of the mammalian enzyme CTP synthetase, which is a pyridoxine-dependent enzyme that transfers an NH2 group from glutamine to UTP to synthesize CTP. This editing event has been recognized in Wilms tumor gene WT1 in mammals and also in the RNA transcripts that encode bombazine-like neuropeptides in amphibians. RNA editing by gRNA. The currently accepted mechanism of guide RNA, directed RNA editing is the enzymatic cascade model originally proposed by Blumet.al. In 1990. Editing occurs in a 3' to 5' direction and is thought to occur in 4 stages: GRNA mRNA anchor duplex formation, cleavage of the mRNA target, the addition or deletion of U residues in the mRNA, and finally, the religation of the 2 fragments of the newly edited mRNA, figure 3.93. Protein translation. One of the fundamental concepts in the central dogma is how the information that is contained within the DNA of an organism in the form of nucleotides is converted to biologically important macromolecules called proteins. Proteins are by far one of the most fundamentally important macromolecules present within the living world, and the process governing the formation of proteins undergoes critical constraints in turning out to have important functions. The process by which the genetic codes in the messenger RNA are built to amino acids, the molecules which are essential building blocks of protein, and then forms a continuous polypeptide chain to form a functional protein is called translation. In this current chapter, we are especially going to focus on how a functional mRNA, after it has been processed, is chosen to be a key player in the process of translation. Its interaction with ribosomes, the most important player known as the tRNA, the various enzymes, and how the entire central dogma is interconnected. Understanding the complexity of the entire process, the following chapter has been divided into various sections for a proper understanding of the key concepts underlying this fundamental process in molecular biology. Fundamental basis of protein translation. Firstly, we must comprehend that the conversion process from deoxyribonucleic acid to M-ribonucleic acid transcription and from ribonucleic acid to proteins translation is very different, and translation is by far one of the most energetically active processes. Translation is quite a challenge for the cellular system to be carried out properly. One of the first questions that came into being was whether there was any scope for direct interactions between amino acids and ribonucleotides in the mRNA. It was studied that contrary to the principle of complementation that exists between DNA template and the mRNA ribonucleotides, there wasn't any specific course of interaction possible between amino acids, their side chains with ribonucleotides. They lacked specificity for any of the ribonucleotides in general. It was hard to imagine there would be even for any of the hydrophobic amino acids as well as aromatic amino acids. Hence, the idea of any interaction between the amino acids and mRNA template for the correct formation of polypeptides and proteins was ruled out. As early as 1955, Francis Crick presumed that prior to the incorporation of amino acids in the respective polypeptide chain, there had to be an intermediate molecular player who could directly interact with the amino acids and hence be able to recognize the 3-letter code in the mRNA. Crick suggested that the intermediate key player had to be RNA simply because the interaction had to follow Watson-Crick base pairing. A couple of years after this assumption, Zamenhof and Hoagland proved that amino acids were incorporated into the polypeptide chain via attachment to a special of RNA molecules which roughly represent 15% of the entire RNA population within a cell. They were subsequently called the transfer RNAs ternus because they assist the attached amino acid on them to be successfully transferred to the growing polypeptide chain. Figure 3.93 mechanism of RNA editing by guide RNA GRNA. The overall process of translation contains 4 key players: mRNAs, ternus, amino acyl ternus synthetases, and ribosomes. These components together help in accomplishing the Herculean task of completing the translation of a code of alphabets into another code that consists of the 20 amino acids. mRNA is the main template that provides vital information about which particular amino acid has to be incorporated. The mRNA consists of 3-letter nucleotide sequences called codons which specify the pecking order of amino acids within the polypeptide chain. The ternus is the key interface that bridges the codons in the mRNA and amino acids. It should also be noted that each of the 20 amino acids which get incorporated into the polypeptide chain is attached to the respective transfer RNA, assisted by an enzyme called the amino acyl ternus synthetase. Every amino acid has a specific turner for itself which carry the amino acid and recognizes the specific codon in the mRNA. The final major player in this entire process is megamachinery consisting of both RNA and proteins called the ribosome. The ribosome's chief function is to coordinate the process leading to the interaction between the mRNA and specific turner carrying the specific amino acid. It further helps catalyze the attachment of the amino acid carried by the turner to the growing polypeptide chain. Our very first focus will be on understanding the roles played by each of the 4 major players in accomplishing the entire machinery of translation. We shall also elucidate recent structural advancements made in the aspect of this machinery and will answer the most important questions revolving around translation in terms of organizational features of mRNA and the codons, structural and key aspects in the interaction between amino acids and ternus and the importance of amino acyl ternus synthetase, and finally how the ribosome machinery helps in completion of this process. Following this, we shall try to understand how the cycle of protein formation and degradation is continued and also the clinical implications of the derailment of this process and new avenues of research in terms of clinical intervention. The genetic code. As we have already outlined the role of ternus in translation in the introduction, we must understand how the specificity of each amino acid being coded by a particular codon and its recognition by ternus is governed. The ternus recognize the codons in the mRNA. Since there are 4 parent nucleotides and only any 3 out of these 4 forms a codon, the total number of effectively possible permutations of these triplet codons is 64. The total number of amino acids coded by these 64 codons is 20. In actuality, there are only 20 amino acids that are incorporated into proteins which are coded by 61 codons, and the remaining 3 codons act as a stop codon which halts the process of translation. The fact that 61 codons code for 20 amino acids tells us that the genetic code is not perfect, in a way that single amino acid is actually coded by 1 or more codons, and this principle is called codon degeneracy. Codons that code for the same amino acid is called synonymous codons. For instance, CUC and CUU are both codes for loose, hence are synonyms. 4 codons UCU, UCC, UCA, UCG code for serine and so on. If we carefully observe the genetic code matrix, we will realize that the initial 2 nucleotides in a codon are mostly the same. The 3rd nucleotide, however, interchanges between a purine with another purine or a pyrimidine with another pyrimidine, and the 2 different codons will code for the same amino acid. Leucine, for instance, can be coded by both CUU and CUC and also by CUA and CUG, besides being coded by UUA and UUG as well. Note that the first 2 nucleotides are the same, and the last nucleotide is either a purine or a pyrimidine. Figure 3.94 genetic code. Note that each amino acid is coded by 1 or more codons, and each of them varies only in the nucleotide in the 3rd position for a specific amino acid. UAA, UAG, and UGA, respectively, are the 3 stop codons. Also, note that the first position in the table is the 5' end of the codon, and the last is the 3' end of the codon. In the genetic code matrix, it was noted that there are 3 codons, viz. UGA, UAG, and UAA, which basically stop codons, do not code for any amino acids which are incorporated in protein, although an exception is discussed later. These 3 codons are not recognized by any internal molecules and are instead recognized by specific proteins called the release factors RF. The 2 established release factors are RF1 and RF2, which are present in bacteria, and ERF1 is present in eukaryotes. Release factors upon entering the A site of the ribosome trigger the peptidyl tRNA hydrolysis, which at that point occupies the P site, eventually releasing the newly synthesized polypeptide. Governing the genetic code. There are mainly 3 rules that govern the genetic code, which primarily determines the arrangement and use of codons in mRNA. The rules are described as follows. 1. The codons are always read in a 5' minus 3' direction. Thus, the coding sequence for tripeptide NH2 glycine would be 5' UH3, where each codon being 5' GGG 3', glycine 5' UU 3' phenylalanine, and 5' CGA 3' arginine. 2. The codons are essentially non-overlapping, and the message does not contain gaps. In other words, the genetic code is continuous. The successive nucleotide codons code for the amino acids in successive order. Hence, for NH2 glycine the sequence would be 5' UH3. 3. The 3rd rule states that the codons that are translated are fixed in a reading frame which begins and ends with the start and stops codon, respectively. The translation usually starts with the initiation codon located at the 5' end of mRNA. Since the ORF is a continuous stretch of the protein coding sequence, it is the position of the start codon which dictates which of the 3 nucleotide reading frames will be used. Cracking the code. In the year 1961, Marshall Nirenberg and Heinrich J. Mattei from the National Institutes of Health (NIH) illustrated for the first time the actual codon. They used a cell-free system that is in vitro conditions to synthesize proteins. They mixed all the ingredients required for protein synthesis, including RNA template, ribosomes, nucleotides, amino acids, other agents which would stabilize the system, buffers, and ATP. They used this system to generate proteins from a pre-synthesized homopolymeric RNA sequence UUUUU, which was synthesized by the use of enzyme polynucleotide phosphorylase, which catalyzes the formation of RNA polymers from the ingredients containing ADP, UDP, CDP, and GDP. The advantage of this enzyme is that it doesn't require any prior templates and can easily generate a polymer that reflects the concentration of the